All right. I think regarding the time that we have, we should start right away. We've waited a bit already. So hello and welcome everybody to MonPlas Workshop. You're gonna see everything about it. You're gonna see how people are fighting against micro and nanoplastics, and we're gonna have a very good overview by Daniel Hill, and we're gonna have the opening by Professor Heidi Ottevaere. So please, Heidi, if you could unmute yourself, the floor is yours.
Okay, thank you very much, Arash. So also from my side, a very warm welcome to this MonPlas workshop. So my name is Heidi Ottevaere, and as Arash told already, I'm a professor at Bibi, University of Brussels, and I'm a member of the Brussels photonics team. Personally, I'm leading a research group on biosensing, where we try to bring added value with optical detection techniques into a variety of applications. And we also try to miniaturize these optical detection techniques into examples are, for example, fluorescence detection of biomarkers for Alzheimer's diseases, dynamics of proteins, but also the detection of micro and nanoplastics. And that brings me seamlessly to the workshop of today. So our MonPlas workshop, where we will give you an overview of the work we do within MonPlas. MonPlas is a Marie Curie ITN training network coordinated by Aston University, in which we have various young researchers tackling different challenging topics within the framework of MonPlas, being micro and nanoplastic pollutions and their analysis methods. So as you heard already, the agenda of today is we will first have an intro on the current challenges of micro and nanoplastic detection by the coordinator of this network, Dr. Daniel Hill from Aston University. And then afterwards, we will have 5 topical topics, which will be addressed by our young researchers, so our PhD students working on those topics. So each time we will have a presentation followed by a Q&A session, during which questions can be raised to our researchers. So this workshop of today is also organized by our B-PHOT student chapter in collaboration with our ESR students of MonPlas. But here I would like, of course, to thank both the B-PHOT student chapter and the ESRs of MonPlas. But I would like to especially thank 2 people. These are Mehrdad, Lotfi, Choobbari, and Natalia Gutierrez Andrade. They were really taking the lead in organizing this workshop and making for you a very interesting agenda. So I hope you will enjoy the workshop a lot, and I wish you a lot of success with the outcome of the workshop in your future career. So, and by this, I would like to give the floor again to our chair, Arash.
Thank you very much, Heidi. It was a very wholesome opening. So I would like to now invite Dr. Daniel Hill to talk about micro nanoplastic pollution and how to tackle this environmental challenge. Daniel, the floor is yours. We can't hear you.
Yeah, so I think somebody put me on. I think somebody put me on mute. I'm probably talking too much. Clear. Excellent. Okay, so yeah, so well, good morning, well, good afternoon rather. And my presentation is about micro nanoplastics, as Heidi said, and it's all about what they are, why they're so difficult to characterize and enumerate or count, and what we're trying to do, you know, how we're trying to develop technologies to overcome this challenge in an EC project called MonPlas. So I'll just give an introduction to the project. We'll talk about micro nanoplastics, then we'll talk about the project itself in more detail, and then I'll just finish with some brief conclusions, and then the more interesting technical parts will come later on after this presentation. So what is the problem about plastics and micro nanoplastics? So the global production of plastics, and of course plastics can take hundreds of years to biodegrade, expected to triple by 2050. And micro nanoplastics have been found in soil, tap water, bottled water, beer even, and of course in the air that we breathe. There's a growing concern about the potential health risks that these particles can produce through either ingesting bacteria on them, which they often pick up in the environment, the environments they pass through, such as wastewater plants when they're in rainwater or effluent from properties or etc., or just through the injury and death of cells through contact. Sometimes the absorption, if they're small enough, by cells, because they believe they can pass through the cell membrane. And so also water companies soon are going to need to measure the concentrations of these microplastics for positive release in water supply and inspectional purposes. And currently they need very sort of expensive scientific laboratory equipment, and also very highly trained and therefore very expensive staff to do the necessary testing. So it's really prohibiting sort of inline sort of low cost testing. And yeah, so these costs are borne by the customers, and if they could be done at all. So we don't really have any reliable or accurate instruments to tell us what we have and how much we have of these micro nanoplastics. So therefore, kind of how big of a problem that they are and where they come from, how much they affect human health, and when water and other resources and commodities are safe to be released for consumption. Because of this, and because the subject is in its infancy, should we say, we put together a successful project proposal to the European Commission call, which turned into a project where we train tomorrow's research leaders, develop the necessary technologies in order to do so, to create these technologies, to create these instruments. And in this project, which we call MonPlas, and there's a video now I'm going to show you. This is the project, beg your pardon, details of the project starting last year and going on till December 2023. And there's the full name of the project. It's called the Training of Early Stage Researchers for the Development of Technologies to Monitor Concentrations of Micro and Nanoplastics in Water for their presence, uptake and threat to animal human life. And I'll just show you now a brief video that the early stage researchers in this project, the 14 researchers have created. Okay, so nice video. Thank you very much again to the ESRs, particularly Atif, for putting that together. Unfortunately, there was some music for that, but because I'm joining you with the Zoom from the browser rather than the app, fortunately, I couldn't communicate. Couldn't sort of you couldn't hear the video, the sound in the video, but maybe you'll get to see it at another point. So anyway, before I share more details of the project, we need to see exactly what are micro nanoplastics and why it's so important that we can study them, and basically why this project exists. And so first question is, what are micro and nanoplastics? Well, macroplastic, obviously easily visible to the naked eye. You can see it on the left of the screen there. Microplastic goes down from, I beg your pardon, mesoplastics 5 to 25 millimeters. Then microplastics 1 to 5 millimeters down. Sorry, 5 millimeters down to about one micron. And these are really sort of below 100 microns are really not visible to the naked eye. So, obviously characterizing at that size is impossible without the necessary tools. That's something we'd like to develop in this project. And then we get nanoplastics, which are even smaller, being below 1 micron. And these are really absolutely invisible. And there's various different shapes. They're more or less spherical particles, can come from, say, cosmetics. These are what we call primary microplastics. These are little plastic beads, but they actually constitute quite a small part. Most of them come from fragments of larger plastic, and the sizes are also the ratio of length to width ratio. Typically, they're about 3 to 1, so typically 3 times as long as they are wide. So they don't normally come in a sort of spherical form. And fibers are known as these ones that have this length to width ratio of greater than 3 to 1. These are very common, and actually it's very difficult to quantify. However, we have to ask ourselves where do they come from, what are they really like, how much is there? And you can imagine that these macroplastics, bags, bottles, face masks, even now with the pandemic, they break down in the environment into smaller and smaller fragments reaching even nanometric sizes. There's various processes involved in the breaking down, and I think one of the ESR groups is going to go more into that. And of course, and indeed, these plastic fragments can undergo many transformations in the environment, and so far and end up very different to what their pristine versions, how they were generated initially. We know that oxidation driven by ultraviolet light can lead to yellow discoloration. And when we think about these things, and I guess we can get covered with bacteria, as I mentioned before, we've seen these large sort of oceanic trash islands, in the documentaries. Well, actually, 94% of those are made by microplastics that you cannot see. So all these things you see in there are the 6% that you can actually see. However, for as much as there is in there, there's actually 30 times more of plastic on the sea floor. Okay, and then there's actually even more on land than there is in the sea. So just to give you some perspective of the size of the problem. Okay, the next slide sort of recaps this and as a key take-home message about micro and nanoplastics. So of those in the sea, 99% of microplastics are at the bottom of the ocean, whilst the 1% on the surface of the ocean, they form the 94% of the plastic you cannot see in these sort of large trash islands. Okay. So, well, why do we care? I mean, about these things are there and they seem to be getting everywhere, but what do we care? Are they harmful? Well, we know that, as I mentioned, the bacteria can colonize the micro nanoplastic surface and modify their density, produce harmful effects if being ingested by superior organisms such as ourselves. We also know that the environmental micro nanoplastics, so those in the sea, the rivers, etc., in the air, they can more easily penetrate cells because they have this sort of biocorona formed from these natural polymers, proteins, fats, carbohydrates, etc. And because of that, they've been linked to various things like cancer, birth defects, and children development problems. And they've been linked. There's no definite proof. And one of the reasons is that we don't have this accurate equipment to characterize and measure. And this is another reason why it's so important to develop these things. So these are the challenges. And principally the challenge here is that, sorry, these are some other details here. you know, they come up. They've been seen in plant roots. They've been seen in the placentas of unborn babies, and they've had a study that showed that bottle-fed babies swallow millions of these microplastics a day, and that food packaging and food is one of the main routes of human exposure to contaminants in plastic. So, but they say the issue is we don't know how much there is because we don't have the accurate, precise characterized equipment developed. And one of the challenges to do so, to develop those technologies, is because plastics are a very diverse group of materials with very diverse chemical characteristics. So it leads to very complicated procedures to sample, prepare and analyze them. For example, your main plastics you would see as plastic recycling, but they're also can be seen as what we call colloquially rubber, such as tires you see on the right hand side. And there are many other types of rubber compounds out there, as you can see in these small images: silicon rubber, Viton, natural rubber, butyl rubber, etc. And then there's paints, for example, they're based on polymeric binders. We have copolymers as well. We have two different monomers unite by polymerizing together. You also have composites as well. So you've got an example of this boat hull. So you have fiber reinforced plastic, or this Tetra Pak type of milk carton here. We have paperboard coated with a waterproof plastic, polyethylene typically. Okay, so these are all plastics. So they're very diverse forms, so that makes it very complicated to characterize them, and therefore develop the sort of reliable and useful tools to characterize them. And it's kind of known as a three-dimensional type of issue because we've got the it's a size, shape and material, or the composition, the type is what we want to characterize. So there's three parameters principally, and before being isolated from their matrix they need to be sampled. And then again, the sampling, you know, depends on the matrix it's found in. So you need to isolate them in different ways, and therefore, depending on whether you get it from the soil, for example, here, or river water, or sea, or even from food, how you sample it can be very different, and also the size that you're interested in. So you might use in an ocean, for example, for something 300 microns and over, a mantra net. If it's in drinking water between 100 and 500 microns, then you might need to use a filtering device, typically with a pore size of less than 10 microns. So this is even before you actually look at it with a characterization tool, maybe a photonics type tool. And of course, when you're doing this, you want to sample extract without polluting the sample. So you don't want to add plastic, and of course you don't want to destroy it either. And plastic is everywhere. So getting your sample contaminated is very likely. So you've got to take some very, controlled steps to do so. You need to wear gloves all the time. You can't use any plastic containers with your samples or micro pipettes. And also, when you're measuring, if you're using high-powered laser, for example, well, that actually might destroy or break down your small particles. So as you can see, the issue of going up here on the top left, the top that you see, you're going from extraction. You start with a large sample, and you work your way across, and concentrating, getting at the exact plastic you need from the matrix that you sampled from. You end up with a very small sample size, and then when you actually want to process a sample and analyze it, you go from a very small sample to these very, very large data sets. So there's all sorts of issues there where you have to introduce machine learning and artificial intelligence to help your algorithms for your analytical tools. So as I before mentioned, we're looking to solve key technological challenges in MonPlas, so we can characterize and enumerate micro nanoplastics. And for this, we have three objectives in MonPlas. We want to improve our ability to detect, trace the origin, determine the toxicity of micro nanoplastics. That's our primary aim. And ultimately, of course, eliminate micro nanoplastics in water. Then we want to add to the limited data on microplastics in water and beverages, develop and standardize a method. Standardization is a key challenge for the initial data taking for nanoplastics, which really has like virtually no data at all. So that's our secondary aim. And with that, because of the nanoplastic relevancy to sort of cell membrane and things and getting to the nucleus, we can study the uptake and effect on animal and human life through toxicological studies. And to do so, we've created this multi and interdisciplinary network spanning various different sectors for which we want to create state-of-the-art detection technologies, applications and methodologies, and subsequently, hopefully, we'd actually commercialize a tool, maybe like an inline characterization tool, and for positive release and inspection of manufactured goods, as I mentioned earlier. So the strategy we're adopting in MonPlas, we apply lab-on-chip technology, previously typically used in healthcare applications, to prepare and to manipulate samples, and then improve detection technologies. And we develop instrumentation in a more systematic and a standardized way. This is something very important, because most people that work in the sort of the genre of microplastics and nanoplastics, they come from marine biology, oceanography, they come from environmental studies, they come from nanotechnology, and they all got different ways of measuring things. And even if they measure the same, should we say, the same thing, whether it be it'll be mass or it will be some concentration, they always use different units, and quite often because of lack of standardization, the data is not comparable. So this is a challenge that we're not leading, but we're certainly being very mindful of, that we want to contribute in some way. And this is to generate greater and far more meaningful and quantitative data. And to do this, we're using this three-pillar approach. We're looking to develop technologies, the application of technologies, and also develop procedures for the use of these applications. And one of the interesting and the key aspects of the project is that we're using the whole of the value chain. So not only got technologists and sort of academic researchers, we've also, or academic entities, universities typically, we also got instrument makers on board, both as beneficiaries or hosting these early stage researchers, and also as partners that are contributing to the training and contributing to provide sites for testing developed technologies. And we also include quality assurance testing end users. And the sort of things we're looking to develop, we're looking at advanced Fourier transform infrared spectroscopy, Raman spectroscopy, combining them with microfluidics. That's something typically done at the Free University of Brussels, for example. And, we're working towards beyond state-of-the-art scientific instruments, and we're using this pyrolysis gas chromatography mass spectrometry as a gold standard, if you will, if you can call it that, to benchmark what we're developing. And because I say that because it doesn't give you all the parameters you need, but it's the most reliable technique for most of the important parameters we have so far, but it's a very expensive and a very lab bolted piece of equipment. As I mentioned before, we're looking for the first reliable detection technology for nanoplastics on real samples, because there's very, very little data on at the moment. And throughout the project, we'll be testing at various sites across Europe, and then looking therefore to map the origins of these micro nanoplastics and working towards toxicological studies. And the actual work packages of the project, the pillars, if you will, as I mentioned before, there's the three pillars. There's the technologies, that's the pillar 1, and the pillar 2 is the application, pillar 3 procedure. And you can see that most of the ESR projects fall under the first one, the technologies, and then there's another 3 on the right hand side that fall under pillars 2 and 3, and because there's some overlap between application procedures, and the sort of 2 pillars are put together. And so these are the people here. You've seen some of these pictures before. Typically, I think you saw the picture of the supervisors and the partners in the right hand corner. There's a little inset. Our project manager, in fact, she's just left us for another full-time position. She was only part-time. Sam Lee. She's done a lot of good work in the project, and we're very grateful for what she's done. And we have a new project manager just begun with us last week. And on the left-hand side, you can see that actually the most important people in the project, and that's the early-stage researchers. And in this case, they're all signed up to do PhDs. It's not obligatory in these type of projects, but they are actually signed up. And we're looking forward to seeing presentations from them today. As you can see, we have a dedicated website there in the top left-hand corner, and considerable social media content. There's Twitter, Instagram, Facebook, LinkedIn, And we have the ESRs. They produce these monthly video logs, and they've got a growing solid interest in our social media. It's a bit late to advertise this workshop on the right-hand side, but that's another example of some of the dissemination that we're doing. So yeah, just moving on to conclusions. I've just got a few minutes left, but there'll be a little bit of time for questions if necessary. So sort of take-home message is that this micro and nanoplastics are a rapidly growing presence throughout all environments, and they have unknown toxicological threats. The MonPlas project, which we are hoping to develop these technologies to tackle these challenges, began on the 1st of January last year. And we hope these applications are capable of the micro nanoplastic characterization and quantification. Almost definitely, if we want to create some sort of, should we say, black box, which you can characterize all samples, even just from one type of matrix, it would have to be a combination of technologies, but even analytical technologies, but that's what we're fundamentally working towards. And the project, even though, of course, we've had this COVID-19 thing going on, which has created various challenges, we have all the ESRs in place. We have results already in the project, even though so most of the ESRs have only been in place less than eight months. And there's publications in motion. I think there's one coming out from my PhD soon from University. We've adjusted our schedule as necessary because of COVID, and we're still on target to reach all our objectives by the end of the project. And saying, ultimately, the project's all about, what micro nanoplastics are out there, how many are there, where are they from, how toxic are they, and can we develop this system for positive release for goods and water supply to consumers, etc. So with that, I thank you very much for listening to my presentation. I'd like to thank the funders of this project, the European Union, and thank you particularly to Jess Folliston from Aalborg University, and some of the ESRs of MonPlas for some of their slide materials. Thank you very much.
Thank you very much, Daniel. It was a great presentation, putting everything in a nutshell. For questions regarding your presentation, we have a Q&A session after the presentation of the ESRs at the end of the first batch of presentations. And if it's okay with you, we're gonna get the questions for you then.
Which time would that be?
That's gonna be around 1:40, 1:50, sorry, 1:50.
Is that your time, 1:50, Brussels time?
Yeah, our time, yeah, CET.
So that's it. Is it about 20 minutes then?
Wait a minute.
I have 12:30 here in the UK.
I have 11:30 here. All right, it's going to be like 1:50, 20 minutes from now. Yeah, of course.
Okay.
Yeah.
Okay. All right. Well, I'll come back to that. I see I'm on vacation just now, but I'll try to.
No worries. No worries.
Okay. I'll call back in 20 minutes.
No worries. Thank you.
Yep. All right. You're welcome. Yeah. See you in a bit. Bye bye.
Bye bye. So I don't know if the presentation is going to go away by itself, or maybe the next person who's going to start a presentation is going to push this out of the way. So we're going to have the first presentation, which is about the origin of these micro nanoplastics and their impact on our daily life, by Yuanli Liu. And then Ali John is going to continue that presentation. This batch is going to be by ESR 11, 12, and 14. As I said, Yuanli Liu, Ali John, and Luca Maurizi. The floor is yours, Yuanli.
Okay, thank you. And good afternoon, everyone. Today, the topic of our group is origin of micro and nanoplastics and their impact on our daily life. And first, I wanted to introduce my group members. My name is Yuanli Liu. I came from China, and I started my bachelor and master study about environmental engineering. Now I study at Aalborg University, and it's focused on the detection of micro and nanoplastics in marine samples. My colleague Ali Chang, he came from Turkey, and his bachelor study is about molecular biology and genetics, and his master study is about biotechnology. Now he studied at Queen's University of Belfast, and his study is focused on the impact of micro and nanoplastics on living organisms. My colleague Luca Maurizi, he came from Italy, and his bachelor study is about industrial chemistry, and his master study is about clinical forensic and sport chemistry. And he is also at Aalborg University, and his PhD is focused on development of new methods for micro and nanoplastics in drinking waters. And here is the agenda. I will introduce the definition, history, and production of microplastics and micro and nanoplastic differing in the environment. Then my colleague Ali will introduce the micro and nanoplastic toxicity, and something about the MonPlas, and also make a summary about the whole topic. First, I wanted to introduce what is plastic. The term plastic is derived from the Greek word plastikos, meaning fit for modeling. This refers to the material's malleability or plasticity during manufacture, which allows it to be cast, pressed, or extruded into a variety of shapes, such as films, fibers, plates, tubes, bottles, and much more. The polymers are made up from two words. Poly means many, and micros means units. Now let's define what is plastic polymers. It means a group of polymers, synthetic or natural, that may be shaped, resolved, and then hardened to retain the given shape. Well, since the second millennium BC, in 600 BC, humans have the knowledge of plastic. Ancient Mesoamericans first perceived rubber into balls, figurines, and bands. In modern times, one of the most used plastic, polystyrene, was first discovered by Edward Seaman in 1839. The first patent for the modern plastic, called Pexin, was taken by Alexander Parkes. Later, he demonstrated in Great International Exhibition in London in 1862. With a quest to find an alternative of ivory for casino balls, celluloid was invented in 1868. Then polyvinyl chloride came in 1872, and cellophane in 1908. The modern plastic era was started in 1907 after the first synthetic plastic patent, which got a US patent for 1909 by Leo Baekeland, which was synthesized by the phenol derived from fossil fuel instead from plants or animals. Industrial practical polyvinyl chloride came in 1926. Low density polyethylene invented in 1935, and industrial practical polyethylene in 1936. One of the most used synthetic polymer, Nano, was invented in 1939, and then polyethylene, the phthalate, was discovered in 1941, and polypropylene came in 1951. These are the most widely used plastic nowadays, but the production rate was not that high until 4 decades of the 20th century. The rapid production was started with World War II. Let's see we change our attitude towards plastic and plastic mass production throughout the last 60 years. If once plastic was seen as a symbol of a rich, developed, In the struggle we live in now, we are more aware about the threat represented by plastic pollution, especially when it comes to decomposition time. We can no more accept or afford to introduce huge amounts of plastic pollutant in the environment. And there are two main types of plastics, thermoplastics and thermosets. Thermoplastics are a family of plastic that can be melted when heated and hardened when cooled. These characteristics, which lend the material its name, are reversible. It can be reheated, reshaped, and frozen repeatedly. Thermosets are a family of plastics that undergo a chemical change when heated, creating a three-dimensional network. After they are heated and formed, these plastics cannot be remelt and reformed. There are around 18 types of thermoplastics and around 10 types of thermosets. The main advantage of the thermoplastic is that they can be recycled, while the thermosets cannot. And the disadvantage of thermoplastic is that they are more expensive, while thermosets are economic. Then I wanted to introduce the plastic production. Plastics are made from crude oil and natural gas, the two beings often found in the same petroleum field. Plastic production accounts for the 4% of the whole oil usage, and nearly 2/3 of its greenhouse gas emissions are produced in the early stage, from fuel extraction to resin production. On the right, we can see a schematic showing the net greenhouse gas emission associated with the production of the most common plastic polymers. So how can we make use of this oil to produce plastic? Oil is a complex of hydrocarbons with different molecule weight and composition. Crude oil can be extracted in different ways depending on the geological characteristics of the field and the type of raw material to be extracted. In any cases, we end up with crude oil, which needs to undergo several refining processes in order to be divided into different parts or fractions. And each fraction is represented by a certain kind of raw molecule. So how can we perform such a fractioning? The effective method is distillation. Indeed, it is possible to separate a mixture's components by employing their different boiling points, which in turn depends on their molecule weight. The fraction serving for plastic production is the one containing the 8 to 12 hydrocarbons. That is to say, hydrocarbons having from 8 to 12 carbon atoms in their chain, which is called heavy naphthalene. The heavy naphthalene molecule then need to be broken into smaller pieces through a catalytic process generally called cracking. Of course, it is easier to get small hydrocarbons from natural gas, as its molecule already possesses lower molecule weights. This is a pretty famous example of mass plastic production. Lego bricks, from a chemical point of view, are pretty complex objects resulting from the addition of different kinds of plastic polymers. This graph presents the production states of all kinds of plastic polymers since 1915. As we can easily see from the image, the annual production of plastic has reached about 7 billion tons in 2015. Now let's talk about the usage of the plastic. First, the highest percentage of plastic production is going for packaging, is 44.8. Then after is building and construction is 18.8. After that is consumer and industrial product is 11.9. For transportation is 6.7. And electrical and electronic is 3.8. And industrial machinery is 0.8. and the rest of it is 13.2 is being used for other purposes. Now let's move to chapter 2. In this part, we are going to talk about micro and nanoplastic diffusion in the environment. As we all know, nature works as a cycle, and the micro and nanoplastics undergo a cycle as well, even if they are artificial. Here we can see we have many sources of micro and nanoplastics, which means that the micro and nanoplastics can enter the natural environment through different gates. Generally, micro and nanoplastics can be categorized as primary and secondary micro and nanoplastics. In few words, primary micro and nanoplastics are those we found in many mass products, such as cosmetics, while secondary micro and nanoplastics generate into natural environment. It is clear that once micro and nanoplastics reach water and air, they can virtually diffuse throughout every ecosystem, and they do. Micro and nanoplastics have been found even in Antarctica, indeed. In the second place, micro and nanoplastics can penetrate living beings' organisms through the food chain, finally affecting us and our dishes as well. Here we have statistics developed by our university, Aalborg University, showing how much we actually lose in terms of macro and microplastics as of 2018. As we can easily imagine, large plastic items such as bags, bottles, and recently face masks can break down in the environment into smaller and smaller fragments, reaching even nanometric sizes as shown in this image. And this breaking process is due to many different factors. Ultraviolet light, oxygen, and physical scratching are only some of them. In this way, secondary microplastics are obtained. Now let's welcome my colleague Ali to introduce the following parts of the topic.
Thanks, Lee. I'm going to talk about the effects of the micro and nanoplastics on our health and their toxicity. Firstly, I want to ask a question that how much plastics are you really consuming in your life? According to the World Wildlife Fund, a report that is published in 2019, 5 grams of the plastics are consumed by human per week, and this means 179 particles per week. So where are those plastics are coming from? First, let me check this pointer thing. So where are those plastics are coming from? They are used for different purposes and in different fields. And such as tires are the biggest area where the plastics are used, which is followed by the pellets and intentionally added purposes. And they are used for the marking and washing purposes. In intentionally added, those plastics are used in the agriculture, cosmetics, detergents, for medicinal purposes, paints, and oil. So those plastics are produced and they came into our lives. But what happens after we use them? They are released to the environment, unfortunately. And what happens in the environment is that those plastics, microplastics, are degraded into small particles by the UV light, natural effects, and biological effects. And during this process, those plastics release chemicals, and they can also absorb the chemicals inside the environment. Another thing, another modification that's happening in the plastics in the environment is that the microorganisms inside the environment, they can form a biofuel around those plastics, and this can cause the biodegradation. After that, after all of those modifications or before them, those plastics are ingested by the many organisms. And by this way, those plastics are coming to our food cycle. So now I want to summarize and talk about the flow and impacts of the micro and nanoplastics. firstly, the macro and nanoplastics are in the environment. Yes, they are. And are the humans and the organisms are exposed? Yes. As I mentioned, those plastics are coming to our food cycle, to our food, by different ways. So how do we expose those plastics? There are three different ways. One is the oral, dermal, and inhalation. And what are the effects of those plastics? For human, there's no direct study. They didn't use any human for the effects of the plastics. Instead of these, they are using cell lines or they are using the animals, model animals for the studies. For cell culture studies, they got the effects of those plastics as the inflammation. Those plastics can cause inflammation. They can cause oxidative stress, lysosomal dysfunction, mitochondrial dysfunction, autophagy, apoptosis, and genotoxicity. For the animal studies, they almost got the same results, but in both ways, those studies are just mostly limited on the polystyrene or polyethylene plastics. But as my colleague mentioned, there are many types of plastics that we are using and that are released to the environment. So more studies should be done to see their effects. Now I want to talk about the main effects of those plastics on the marine ecosystems. These effects can be divided into direct and indirect effects. For direct, those plastics are taking space up in the guts and digestive system of those organisms. And the other effects are the physical disruptions. They can reduce the dietary intake. They can cause energy depletion, oxidative stress. Also, they can slow the metabolic rate. Those studies are mostly done in the mussels, oysters, and urchins. And another effect is that they can inhibit the growth. And lastly, those plastics can reduce the reproducibility of those animals, of those organisms. Another thing that I'm going to talk about is the additives, actually. During the production of those plastics, different chemicals are used for different purposes. And when these plastics are released into the environment, they release those chemicals, or also they can absorb the chemicals which are dangerous for the health. So on the, let me open the pointer first. On this part, you can see different plastics and the chemical additive that is but for the purpose of those chemicals in this production of plastics. Those additives can be used for the antioxidant purposes. They can be used as flame retardant, plasticizer, and stabilizer. And the dangerous elements, these dangerous substances inside those additives are the phenols, phthalates, anti-flame retardants, and also the phenols. Again, same. You can on this part you can see the additives, chemicals used, the flame retardants, phthalates, nonylphenols, and the bisphenol A and erganox. The main effect of additives are on the endocrine system. They can disrupt the endocrine system, and those disruptions can cause many health problems inside the organisms. I'm going to talk about this later. And also in the middle, you can see the purpose what for are they used for, again, antioxidant to reduce the flammability and for plasticizer purposes. Firstly, before talking about the effects of those additives, I want to talk about how they are affecting, how they can disrupt the endocrine system. Those chemicals can decrease or increase the response, the cellular response to any factor or any effect. Either they can mimic the hormone. By this way, they increase it. Other way is the blocking the hormone. They can also bind to the receptor, and by this way, they can affect they can block the hormone to attach there. So you won't get any response if this cell or the organisms face with any danger or any factor in the environment. And according to some studies, they tried to see the effects of those EDCs. And they saw the effects in the reproduction system. Also, they saw the change in the response to psychological stress and growth and development problems, and other one, cancer. For reproduction, as I said, those EDCs can mimic the hormones. They can block the effects of the male and female sex hormones. And the other thing is that what they saw is that those EDCs can have the neurological and can cause the neurological and behavioral changes. And also they can reduce the ability to handle stress. Another thing, another effect of the EDCs is the growth and development problem that I mentioned. The high exposure to these EDCs during the gestation can lead to the low birth weight. They can alter the development. They can disrupt the sexual development, and also they can weaken the immune system. And the other thing, and one of the important things, the important effect of those EDCs is the cancer. Exposure to estrogen or androgen mimicking EDCs can promote breast or prostate cancer, and or those EDCs can interfere with the hormone cancer therapy. Another study showed that the prenatal exposure to some EDCs may alter the mammary gland development and increase the breast cancer risk later in life. So I'm going to talk about what MonPlas is going to do in general. Firstly, as MonPlas Consortium, we are aware of this danger. As you can see on the right graph, the production of the plastics are increasing dramatically, and every 10 years, the size of the plastics production is doubling. As you can see, in 2010, it was around 300, and on 2020, it's around 500, or we can say 600. As you can see, those increase are, we don't know the real effects of those plastics, even there are some studies. But as those plastic pollution is increasing, with the production of those plastics, we should investigate and try to see and try to prevent those pollution. As a MonPlas, we are working on the detection of the plastics in the water. And firstly, they are working on water now, and they will pass on to the beverages and food. And also, we are trying to trace the origins of those plastics in the environment. Other thing that we are working on the effects of those plastics on the living organisms. What is the advantage of the MonPlas is that we are from different industries and different universities, and we share the knowledge, we share the information, we share our experiences. We can always consult each other, we can always talk, and we can get trainings from each other. So that's why the MonPlas will help us to find better solutions for this plastic pollution. As individually and personally, what we can do? We can refuse the use of the plastics. For example, instead of using the plastics, you can use the paper bags, or you can find another alternative way instead of the plastics. Other thing that we can do, we can try to reduce the plastics that we are using in our daily lives. The other thing, and the third thing, is that reuse. Instead of using the single-use plastics, we should be aware and that we should try to use those plastics many times in our daily life. Lastly, we should recycle. No matter the plastic we are using, we should try to recycle. As the plastic production, actually, the main problem is caused by the not recycling those plastics. So we should take care and pay attention to this point too. To summarize, we first talk about the plastic story and production. Secondly, we talk about the diffusion of those macro and nanoplastics in the environment. And thirdly, I talk about the effects of those macro and nanoplastics on life. And secondly, I talk about MonPlas. I want to thank you all for listening. And if you have any questions, we would like to answer.
Thank you very much, Ali, and also Yuanli. So now the ESRs, Yuanli, Ali, and Luca will be forming a panel, and I hope Daniel is back and Heidi is here so that you can all answer the questions that our audience might have. So guys, if you have any questions that you haven't put in the chat box, you can also raise your hands and ask your questions yourselves. But I'm going to start with the questions in the chat box, and you guys, any of you who has the question, who has the answer, can actually weigh in. So first is from Florian, who's working in Horiba Scientific, showing interest in your consortium and seeing some points that they can actually collaborate. So they're saying what technique you're using for nanoplastics. I think he means by like optical techniques like Raman, FTIR, and so on. So can you guys actually have an answer for that?
Actually, our other colleagues, they are going to talk about these methods, so maybe it's better to ask them after they present.
Yeah, that's a good idea, because he actually is asking about their limitations.
Yes, can you hear me?
Yes, Florian.
Yeah, yes, it's me. Thank you for this presentation. So yes, I am working for the company, for the Horiba Scientific. So we do manufacture scientific instruments, and we are really involved in this hot topic of microplastics. And so far we are using micro Raman because it's it has how to say better specification than FTIR. We can go below the 30 micron. We can analyze really microplastics. But to analyze nanoplastics, maybe we should think about a combination of Raman and another technique, which we do manufacture, AFM. So that's why that's my question actually. Because you were talking about nanoplastics. So today, which technique you are thinking for analyzing at nanometer scale these plastics?
Florian, I think if you are still here after 3, ESR 1, 7, and 10 are going to talk about these things, how they actually detect these kind of things using different tools like Raman spectroscopy and so on, okay, like NPR. But I don't know if Daniel wants to give you a short answer, if he has any.
Yeah, I can do that. There's various techniques we're using in the project, mostly revolve around SIRS and the variants of SIRS. Okay. I think you'll see later on in the more sort of in some of the other presentations, you'll see more details on that. But you know, we're pretty much earlier on. But the idea is that we should, you know, use techniques that we've developed in lab on chip for lab on chip platforms, and then use those to you know, prepare and to manipulate samples in order that we can use typical sort of SIRS platforms. AFM is a possibility, but as maybe you know, I mean, I've worked with them. Excuse me, I've worked with them before, and it's not the sort of technique you would use to quantify in a sort of study or a commercial control process. If you wanted to use it as a research tool to say, hey, we can look at some samples and we can get some results, then sure, they can be combined, and you can think of using NSOM, for example, with SIRS as well.
Yeah, but with SIRS, you will still have the limitation of the size of the laser spot. So even if you are using SIRS, if I'm not wrong, even if you are using SIRS, yes, your Raman signal will be enhanced, but you will still have the limitation of the size of the laser spot, and maybe you are using a green laser. So in a minimum, you will be able to detect 10 microns, something like this, or around 10, a few microns, but you will not reach the nanometer.
Well, I think it all depends how you couple out the light, because obviously fundamentally what we're interested in is coupling the light to the structures, the plasmons of the structures, and then it's the interaction of the near field, isn't it, with the plastics, and then it's all a question on how you couple the light from the near field, and also, you know, how strong it is. I mean, we're not looking to necessarily resolve you know, if it, how can I put it? The detail is in the microfluidics. Okay, if you can separate the particles out so that they're they're largely, so they're isolated to, you know, and you scan an area which is less than the you know, the diffraction limit in size, 500-600 microns with a micro Raman, then, you know, if you get a big enough signal, which is the key part, then, you know, you get all the information you need. Okay, obviously, if you have several particles, you're not going to be able to discern, are you?
Yeah. Okay.
Thank you, Florian, for your question.
Yeah. All right. Thank you.
Thank you, Daniel, for your explanations. So the next question is from Luca Schifano. So he's asking that, okay, Alison said in the end of his presentation that, oh, okay, we can repurpose plastics, we can recycle them. Is there any technique that MonPlas is looking into for reduction, transforming, or avoiding the production of micro nanoplastics in the environment?
As a MonPlas issue, we are not working on that. The last part that I mentioned is a kind of individual responsibility for each of us. For example, if you are using plastic and you saw the recycle bin, just throw it, if it can be recycled. So it's not about the MonPlas issue, it's just for individual responsibility.
Yeah, Ali's quite right. In the MonPlas project, there's no direct, you know, we're not going to affect these things directly. Okay, it's indirectly. If we have these tools, then we can characterize and we can quantify where we see these particles, so we get a better idea where they come from, and then we can produce more accurate and precise and larger volumes of data, and then the environmental scientists can use that as an argument to say, you know, I don't know, these water treatment plants need to change their processes because this is the evidence, yeah? Or producers of washing machine need to change the filters they put on their washing machines, okay? That's our indirect influence.
Brilliant. So we have another, well, interest from Daniel Oppe in your consortium and in your work. But the question is if nanoplastics have been chemically characterized. I don't know exactly what it means. Daniel Oppe, can you actually unmute yourself and maybe explain a bit?
Okay, good afternoon.
Okay,
good afternoon. Thank you for the presentations. Yeah, I've followed, and then I think the next presentation addressed that question already.
All right,
so
all right.
I've been able to see that it has been characterized.
All right,
yeah.
All right, perfect. And your question is already answered in the future. So another question from David Surdeco. He has a question about your goals. You mentioned that you want to detect plastics in water. Do you mean water bodies like in seas, lakes, drinking water? Or where do you want to detect them actually?
Lily, maybe you can answer.
Yes. My PhD goal is to detect the concentration of micro and nanoplastics in marine environment. So it's from the sea, and my colleague Luca is looking for detect micro and nanoplastics in drinking water. So it's actually looking for the body. And I think next group will also do some presentation about how to do the sampling and how we do the sample preparation in our lab, and then finally how to analyze the micro and nanoplastics, and then we get the results. Yes.
So maybe I'll just, yeah, hi. So yeah, I'm David. I'm a PhD student at the University of Twente, and actually my side project is related to removal of microplastics from water, so to degrade them in some way. So that's why I'm interested in it. So when I started searching for information about contamination when it comes to microplastics, what I noticed that there's not really that much information, right? So I found some, but it's very scattered. So one thing is, for example, some researchers report the contamination of microplastics, you know, in a particular water body, they give you some data, but it's not precise enough. So for example, they only give you a very wide range of the sizes of microparticles, right? Then another researcher does it in a completely different way. And there's no like unified method to do it. So how let's say reliable your data would be, how exactly do you plan to do it? Because for my research, it could also be very useful data.
Yes, this is a particular challenge which I flagged at least once, I think, in my presentation. There is a general lack of standardization of methods and approaches, not just in terms of when you analyze a sample and what you measure, but also how you actually take a sample from a matrix and how you process the sample. Because it's kind of a bit like the Wild West at the moment, yeah, because people come from different backgrounds, oceanography, marine biology, other types of environmental science, nanotechnology, civil engineering, you know, they're coming from everywhere and everybody's using different methods, and there's no kind of agreement yet on exactly what should we measure. Some people measure, you know, they do a breakdown of particles based on their mass. Some people will do a size distribution of particles. So there's many different ways, and it's not really within our remit of this project to develop any standardization, so we can all generate useful and comparable data, which I believe is what you're alluding to. However, we need to be aware of other activities and projects that are underway to developing this standardization.
Okay, thank you very much. And also, actually, that may be relevant for you. I'm not sure if you are aware, there's a foundation in Netherlands, here where I study. It's called Plastic Soup Foundation. And they are also very interested in that. So, yeah, okay, since you know it then, yeah. So, thank you.
All right. Okay. So, thank you, David, for your question. Then we have our last question from Yusuf. Sorry for the rest of questions. So, thank you very much for your presentations. Question is, are there any biochemical agents that can be used to mitigate the effect of microplastics on hormones? You know anything about it? Just
yeah, actually, I got actually what Yusuf is meaning. Yusuf, thanks for the question. But actually, I couldn't see any study. Also, I'm not a chemist, I'm a biologist, so I didn't look actually, to be honest, to any study for this purpose. But yeah, this can be investigated or looked for it. Now, mostly nowadays, they just focus on the effect, not instead of to find solution, how to compensate those chemicals. So I don't know much about that. Sorry about that.
Yeah, I think fundamentally this is a socioeconomic issue. Until we can actually produce hard data to say, you know, this is the damaging effect. This is how bad it's doing to animals and humans, etc. That it's very difficult to argue that they should limit the production. Yeah, so I think that's why the focus is first of all to measure how much there is, what the effect of a certain quantity is, and then when we have that data, then we can have a bigger, stronger push for people not to produce so much, or to to neutralize what is there.
Yeah. So perfect. Maybe we can close this section and go to manipulation of manipulation technologies for these micro and nanoplastics. So there are 3 ESRs that are gonna be in this section: ESR 3, 4, and 6. Selim Tanrıverdi, Martim Costa, and Clementina Vitali. And the representative for this group is gonna be Martim Costa. So if you can unmute yourself and share your screen, I'd be grateful.
Thank you very much. Let me share my screen. Let me know if you can see everything, please.
Yes, everything perfect.
Yes, excellent. So everyone, welcome to the second more technical presentation of MonPlas, focusing on the micro nanoplastic manipulation technologies. And so I'll simply tell you a story about the things that we are working on, some of the state of the art, and in a bit what you can expect from our project in this regard. So firstly, a small introduction regarding us. So Clementina Vitali, an Italian lady, so she's a master in food science and human nutrition. She's part of our team, as myself and Selim are, and she will be analyzing micro nanoplastics in water, beverages, and food. Myself and Selim, we work in KTH, in SciLife Lab, Stockholm. And while I'm a master's in biomedical engineering, Selim is a master's in nanoscience and nanotechnology. And in a way, we are working together in microfluidics to find, let's say, interesting solutions that can help us take a sample, prepare it, and remove everything we don't want. And so let's simply give a small introduction about our institutes, because it might be relevant for some of you. So in terms of Clementina, in the Wageningen Food Safety Research Institute, they wish to explore the potential of nature to improve the quality of life. And what this means is simply, well, having a lot of, let's say, food applications and concerns, it's necessary to understand where contamination can come from and where toxins can come from. They have 3 business units, and one of them focuses precisely on this. Secondly, they have a very, let's say, methodology development way of looking at things, and then of course they apply it in a 50-50 way, let's say. And taking this into consideration, on one hand you have Clementina, our ESR, and her promoter Michelle Nielsen, and finally her supervisor Ruth Peters. And together with also 5 researchers, they aim to make this a reality, being able to treat their samples in the best way for the purposes of microplastic research. In terms of myself and Selim, we come from KTH, as I've already mentioned, and in our institute we work with many things, but we were firstly focused on nanobiotechnology, as you can see. So paper microfluidics, droplet microfluidics, you can see some very interesting videos here of precise control of fluids, inertial microfluidics, as you can see. And so what I want to convey to you, even though this is medical, is that many of these technologies, droplet microfluidics, point-of-care diagnostics, let me just take here a nice pointer so we can draw a picture very clearly for you guys. So single-cell sorting, all of these technologies have huge interest nowadays, and many of them, if one is smart and let's say wise, can also be transposed to the environmental sector, and so microplastic separation, in which myself and Selim will be working. And so this is where we work, in SciLife Lab, if this will allow me, of course. So SciLife Lab, beautiful building, circular in shape, I think everyone can see that, and KTH, beautiful, the Swedish weather, as you know. So in terms of the agenda, so it's easier for me to tell you the story from the beginning, step by step, so none of us are lost. This sounds a lot more complicated than it actually is.
Excuse me, Martim.
Yeah.
Can I think your slides are not moving from slide 3, or are you on slide 3?
Oh, that's very strange. Let me try to screen share once again.
Yeah.
Do you see it now? Which slide do you see at this time?
Now it's black.
Well, I will simply do it the hard way and share my entire screen.
That might be useful.
Do you see it now?
It just says Martim Costa has started screen sharing. Anybody else can see it, or I don't know if it's my problem.
Yes, I can see that too.
So you cannot see. This is quite inconvenient. Allow me to try and find a solution. Share screen.
I think sometimes it takes some time for Zoom to load the presentation.
Yeah,
so if you just wait a little, it might work.
Okay. I'll try to once again share. I am vexed.
Do you want to maybe close the session for yourself and come back?
Perhaps that would be useful.
I don't know, like, yeah. Oh, it's here.
It's here?
Yeah. Now I can see it. Perfect. 4. Now it's on 4.
4, 5.
5 now.
Okay, where did you lose me? I can do it again. It's no problem.
You were talking about the KTH and the circular architecture.
Okay. Okay, very well. So now you can see the slides, the videos, and so on that I was talking about, perfect. The droplet microfluidics, the inertial. And so in the interest of time, I will not say everything again, but these technologies in a way can be applied for the environmental aspect. And these are simply pictures that I was describing to you of the place where we work. Now, if you are still following me, in terms of the agenda, firstly, we will start with sampling. So how can we classify micro and nanoplastics, so MPs and NPs? Then how we can analyze them in an elegant way so we can have a little more information about them. Thirdly, how we can look at it from a contamination point of view. Fourthly, we will dive into the world of microfluidics, which we will take step by step to make sure that we're all in the same page. And finally, two types of separation methods in precisely microfluidics, one of which is passive, another is active. And finally, we will conclude it and wrap everything around so we're all satisfied with the result. So, in terms of micro nanoplastic classification, observe that one type of classification is simply size. So we can look at anything, we look at our natural world and things have different sizes. So this is a very intuitive way of doing so. You can see that there are many different dimensions of microplastics, which allows us to divide them in different terms. Macroplastics, large microplastics, a normal microplastic, and nanoplastics. This is one way of looking at it. And not all of our contaminants are, let's say, perfectly round shaped. So they have different dimensions. The way we choose to evaluate this is simply if you pick any particle, you look at the biggest dimension of it, and then that's the dimension that you work with to categorize it in terms of its size. Another type of classification is shape, because unfortunately, this field also had to be complicated. So we don't only have spheres, we have fibers, we have flakes, we have foams, and we have to take this into consideration during our science, especially when working with separation, because one thing is trying to separate a sphere or an egg, for example. Another thing is trying to catch spaghetti. So we need to take that into consideration and view how this can affect our systems. Next, origin. So origin is, how can I put this? You have two types of origin of microplastics. You have primary and secondary. Primary are particles that come off from a specific function, in the sense that, for example, when someone puts makeup on, there are microparticles that can be leached off when we simply wash it. And this goes into the environment. This is also an issue with pharmaceutical and medical items. Another type of origin is secondary. So secondary is simply you throw a bag, you shouldn't, but you throw a bag into the sea, and with UV and other types of natural erosion, it will degrade and slowly become a microplastic. Maybe not so slow as we'd like though. So continuing, how can we analyze them? This is an excellent question. So observe that when you sample something, so you go into the natural world and you take something, you need to prepare it, simply because we are dealing with such small things that it's not particularly evident how we can go about this problem. However, we still want to quantify them, how many microplastics there are. We want to know their size distribution, if they are big, if they are small, if they are nano, if they are micro. And finally, identifying them. So we know which type of microplastic it is, because there are many, as you've already seen from the previous presentation. They can be polyethylene, polypropylene, and this is also important to know. And so a way that we can look at it is that they have different densities, and you'll see that this might be quite interesting to look at later on. In terms of sampling, so as you deal with sampling, you have to understand that when we take sampling, when we evaluate a new system, the complexity is different. So one thing is looking at water, clear water, and trying to analyze it. Another thing is looking at sludge, oil, and the like. And another thing is looking at, for example, a living organism, which is extremely complex and ever-changing. And so our technologies need to take this into consideration, especially in the beginning part, in sampling. And so having this in mind, we have two types of sampling. Oh, my laser pointer has disappeared. Let me take it once again. You have bulk sampling. So let's imagine you're making spaghetti. You have a big pot. You take your colander and you put your entire pot through the colander, and you can take the spaghetti out. So this is called, in a way, bulk sampling. You take a big thing and you process it entirely to remove your spaghetti. And an example of this is simply shoveling. It's the good old shovel. You take everything out, you bring it to the lab, and you look at it. Another one is volume-reduced sampling. And to make it clearer, it's simply imagine you're boiling potatoes and eggs. So you simply you want to take the boiled egg out because you don't want it to be overcooked. So you take it with a spoon, but you don't want the water to come with it. So you try very carefully with all the skill in your being and fiber to take it away. And then you recover only the egg, which is what you want to look at. This is volume-reduced sampling, roughly speaking. So this can also be applied to microplastics. There are many types of sampling in our world. These are pictures from Aalborg University. And to take water from the natural world, as we can see, we need to take into consideration the mesh size of our nets and what type of technologies we want. What do we want to study with this? Do we want big particles, small particles? Do we want things that get stuck with high speeds of the boat? So there are many parameters that can be manipulated, not only from an experimental point of view, but also from a hardware point of view. And my personal favorite, manual net sampling, which requires some strength to do and is a bit more manual. So this is one way of doing it with our great technologies. Another way of doing it, which is not only, let's say, liquids, so marine water, we can also do it by physical dirt, let's say shoveling, and also clawing. So on one side, so to the left side, look, this is the piston borer. The piston borer goes down into the earth and takes out an entire cylinder of soil. So you can imagine this has a lot of value in being able to study not only temporally, but also in terms of space, any pollutant that you might have. Behind here, you have a map of Aalborg and some samples that they've taken, as well as their geography. So on one hand, you can have the trusty old shovel. On the other hand, you can have these huge metal claws that we've seen many times. And so this is simply another way of taking a sample that's not watery. It's soil, and this needs to be, let's say, processed in a different way. And then when you finally take your sample, imagine you take your sample, it has a ton of stuff. In the water, you have algae, you have microorganisms, you have oil. So you need to work with it somehow in a way that you can isolate the microplastics from everything else. And there are many ways of doing this, but the important part is that you treat your microplastic or your analytes and you don't ruin it. This is a very interesting image that shows many different types of acidic treatments, alkaline treatments, oxidative treatments, and enzymatic treatments. And you can see that the black dots are the effectiveness, and the white dots are simply a measure of how much the microplastics were affected by this treatment. Enzymatic treatments are quite successful in this regard, and in a perfect world, we would have all treatments work in this way. But this is, of course, a field in development. But you can see that this is a challenge. There are many ways of doing it, and many different types to pick up the problem, but it's not obvious exactly what is best for each particular situation. And it's precisely because of this that this area has a lot of interest in terms of the MonPlas development. Now, as I've given you the spaghetti example with the colander, you can filtrate stuff. So everyone knows filters. You just put something in, the big things stay, the small things go through. There's nothing particularly complex about it. You mess around with the mesh sizes of the filter, and then you can have different results. Secondly, you have sedimentation. All of us have seen or done or made smoothies. So, and you can see that different densities, if they are not mixed, they simply deposit themselves in an organized manner by density. And this is something that you can do with to separate your microplastics from other solids using salt solutions. So these are two very simple ways we can look at in terms of potential applications. And so having this in mind, like in the lab, much like a kitchen, we need to be careful what we put in the food or on our experiments. So prevention is necessary. These are simply some tips and tricks that you can use in your own lab work to prevent any plastics from contaminating your sample. Because if you're putting plastics in your sample, you're not really measuring what's in the sea. You're measuring what's in the sea plus some error. So you can set up a clean room. Clean rooms are always beautiful to prevent any kind of contamination. You can also, let's say, evaluate the purchase of a dust box. You should take into consideration your lab coat due to its cotton, let's say, how can I put this? If you wear cotton, it's a good thing. But if you wear synthetic fibers, especially as you wash them off, they can come off and contaminate your sample. Secondly, lab materials, while many of them are made in plastic and it's a necessary evil in some regards, it's something that we need to take into consideration. So using glassware and washing it is of utmost importance. And finally, you should run blanks to make sure that you know exactly how much you're potentially contaminating. So having this in mind, let's go into microfluidics. And the name is scary, but it's not very scary in truth when you know it. So microfluidics is simply, you have a pipe. So imagine a big pipe. Now make the pipe very small. And when I say very small, it's the breadth of your hair. So pick up your hair, right? Pull it. And the thickness of your hair is the thickness of the channels that we work with. And this is advantageous for many reasons. It's a small volume, so you don't need many reagents to try different experiments. You don't take very long to heat it either. And so you can test many different conditions quite quickly. This is beautiful in itself. And many times, depending on your material, you can use microscopy to view in real time what your experiment is doing. Many areas of science use this, and this is also an excellent tool for this environmental area. And I'd like you to keep in mind two things. We are working with very small sizes, and we are working with laminar flow. What does laminar flow mean? I'm glad you asked. So you have two types of flow in all fluids. You have turbulent flow and laminar flow. Laminar flow simply means that your fluid does not mix. In very small pipes, your fluid simply goes forward like an arrow, and unless something acts upon it, nothing happens. So it just keeps going forward forever. The fluid in the middle goes faster than the ones away, simply because the ones away have to interact with the wall of it, in roughly speaking. So faster in the middle, slower here. Turbulent flow is what you see in big pipes. It's basically what Britney Spears said in her 2011 pop dance album, Femme Fatale, in that we are dancing until the world ends. This is excellent for pharmaceuticals. So you're doing chemical reactions and you're mixing everything up. However, for microplastics, this is not very useful. We want to be able to separate them, to enrich them, so we can send them to the analytics guys. And this behavior can, in a way, be modeled by the Reynolds number. And very simply, the Reynolds number puts the density, the velocity, and the size of your microchannel, which is very small, remember. So it's a very small pipe with the viscosity. And if this L, if this length is very small, then your Reynolds number is very small. And so you have laminar flow. This is where we work. So taking this into consideration, we can apply this to something called lab on a chip. Lab on a chip are simply microelectromechanical systems, so MEMS for short, that using microfluidics can miniaturize a laboratory. So instead of having a huge laboratory, you can have it all in a small chip if you design it correctly. And this is a technology used in many areas. It's cost effective. If it's designed well, it works, it has sensitivity. And so it's something you really want to have if you have the brains to do it. And so there are many different types. So you can use it for cell cultures, food testing in terms of toxic, diagnostics like COVID chips, but also environmental applications. And this is where MonPlas comes in. How can we use lab on a chip solutions and microfluidics to help with these microplastics? Well, That's a very interesting point because microplastics differ in size, shape, density, electrical charge, acoustic impedance. And so we can dance in many different styles here, and that's the beauty of it. So the first type of separation that I will tell you is passive microfluidic separation. And this is simply separation by design. Now, this looks very complicated, but just bear with me, step by step. As I told you, in the middle, the speed of fluid is very quick. So when the speed is here is very high in the middle, it will push particles out like a wake, yeah? So your particles that are in the middle, they will be forced by the wake of the fluid to go next to the wall. However, they cannot go through the wall and the wall pushes them back. And what happens if you leave a small pipe that's lengthy enough, undisturbed, these particles will assemble spontaneously in a lined manner. According to the type of your microchannel, so let's say circular, they assemble themselves in this equilibrium position. And the force that keeps them in place is one we will call the net lift force. If it's square, it's like this. If it's if it's rectangular, you have another type of conformation. But then you'll ask me, but Martin, these are a lot of particles going at the same time. And if you work in photonics, like my colleagues, they'll tell me, Martin, would you take it wrongly? Would you take it too hard if I wanted one particle, just like an airport? You know, you go into the metal detector, one person by one person. I also want my particles to go this way because I want to identify them. So would you hold it against me if I wanted you to find a solution that the particles go one by one? Even though your solution is already very good and divine and a beauty of nature, we want something better. There's no problem. This has already been invented. And this is invented simply by a microchannel such as this. And so this puts a centrifugal force, which we are all aware of, into the channel. So everything is mixed. As they go through, the particles assemble themselves, and then by putting different outlets, spaces for them to leave, they can simply be separated by size in this way. It's deterministic and requires no actuation. And what happens is that because of this centrifugal force, the fluid will create these vortices. And these vortices are called Dean vortices. And they make the particles want to move along the vortex. This is a microchannel that is coming against us, right? It's flowing against us. But as it's flowing, the particles are being torn in this way. However, because of the forces that I told you before, the net lift force, they also don't want to be in the middle because it's going super fast. It's scary. They want to go next to the wall, but they can also not go too close to the wall because they'll be pushed back. So you have a competition between the Dean force and the lift force in such a way that by size, the equilibrium position of these particles will be different. And that's why big particles stay here, medium stay here, and the smaller ones go in the middle. And this is something natural. This is like my mother, beautiful and elegant, and it's something we're going to use for our projects. Another type of passive separation is the use of filters. We all know this. So this is a very nice paper by Udit Pant. And one problem with filters is that they get clogged, right? And we see this even at the macro scale, even though at the micro scale and the nano scale, the physics are a bit different, as you've already seen. And so what they did is simply they put pillars and the big particles stay and the small particles go through. However, given enough time, these filters will simply be blocked. So what did they do? They vibrated. Oh, there goes my mouth. They vibrated. So they simply punch the microchannel, it wobbles a bit, and the small particles can continue to flow. So this was a smart idea to fix one of the problems with filters. But of course, there are advantages and disadvantages to everything because your large particles get stuck here and you can't remove them easily with this kind of system. So another thing we have to develop. Finally, you have something called deterministic lateral displacement. Of course, what I told you here in no way exhausts the amount of technologies that we have, but they're interesting ones to take into consideration, even for your own research. And what happens here is that by clever engineering of these pillars, you can make it so particles leave or stay in their flow lines, because this is laminar flow, remember? So with small particles, they can simply follow their flow line. So as you can see here in slight gray, they flow and they go out in the one lane. But if they're big and they want to follow the two lane, for example, they will come here and be stuck, but they are still pushed by the flow. So they will keep going, and with enough pillars and clever engineering, you can simply displace them to be collected in another outlet. So this is also a very interesting way of separating particles by size. Now, active separation. As we've seen, we can... Why are you going without me? Don't have a mind of your own. Anyway, so as you've seen, we can design a microchannel and it will do something for us. And here the labor is in designing. But we can also design a microchannel that, aided by a system, separates particles. So an example of this is acoustic separation. So observe this pipe, very small pipe, a basic microfluidic channel with water. If you put a sound wave through it at a particular frequency, you can create a zone of no pressure in the middle. And what do particles do when confronted with high pressure and low pressure? They want to go to the place where there is no pressure. And so in this quaint simulation, you can observe quite obviously that the particles, colored, they follow almost exactly the no pressure nodes in gray. And this happens in real life as well. We can create systems that actually do this. Of course, particles will behave differently according to their density, according to their size. And the force that actuates here is called, let's say, acoustic radiation force, and it impacts bigger particles more than small particles. So through engineering of microfluidics, we can indeed send one particle one way and another particle another way. Of course, because we are dealing with big amounts of fluids, we also want to have high throughput. We need to be able to use high amounts of fluid because microplastics are small, right? But of course, the disadvantage with this technique is that because sound waves, as they go through your materials, they dampen and they heat. So, and when you heat, the way sound travels in any medium, air or water, for example, changes. So this is something to take into consideration. Next, observe even at macro scale, using sound waves here by Andrade, you can levitate particles, plastics. This is magic, for lack of a better word. And secondly, in this paper by Wu et al., in which they deal with blood, it's not microplastics, but the sizes are similar. By using ultrasounds, he can separate blood from their laminar flow lines into the middle. And so finally, this is the last point before I wrap up my presentation, which is called field flow fractionation. And what this means, you have a pipe. It's not very complicated. The name is just a bit strange. It's cool, but it's a bit, let's say, cumbersome. So you have a pipe, you push your particles down using a thermal gradient, acoustics again, magnetic, and the particles, according to their size, because of physics, they will migrate up again because they want to, they want to go back to the balance of the position. They will migrate up, and the particles that reach the center quicker will simply be pushed forward. When you have very small particles, they are dominated by Brownian motion. Brownian motion is simply how particles vibrate with temperature when they're very small. So particles that are smaller vibrate more, so they go up into the middle. Secondly, in steric mode, so it's intermediate, bigger particles, because they're larger, their center of mass is closer to the middle. So they catch the wake and they go faster forward. So finally, taking this into consideration, the hyperlayer mode is not very different. Bigger particles are pushed harder from the wall, they go to the middle, and they are expelled quicker. So this is an intelligent way to displace these particles and separate them. And so I give you now a wraparound of these technologies, and I will show you the conclusions of what can you take from this presentation. Well, what does this all mean? So in terms of classification, we need standardized protocols to be able to cross-reference with different people. also working on this subject. In terms of sampling, there are many techniques, as you've seen. There are many types of preparation that you can do, enzymatic and so on, as I've showed you. So more need to be developed so we don't compromise our microplastics when we do get them. Finally, each microplastic having its shape, its size, its density, maybe benefits more from a certain type of technology than another, as I've shown you. So this is also a point to brainstorm as we move on with our projects. And finally, we want a solution in MonPlas that you take your sample and you can give an answer and send it to analytics so they can tell us what kind of plastic it is and what to do with it. So thank you very much for your presentation. I'd like to thank my two colleagues that helped me with this work, so Clementina and Selim. And you have here our email contacts, and I'd like to thank the MonPlas Consortium and our funding of Marie Curie. Thank you very much for your patience and interest in my presentation.
Thank you, Martin, for your presentation. We're a bit behind, so I'm just gonna quickly go to the questions. So the first is from Danielle. So what factors do you see for the degradation of plastics? Both of your colleagues can actually also weigh in if they want.
Let me, I'm reading the question. Yes, this is a good question. Because plastics assort themselves differently in, let's say, ocean, seas, some will be at the top, some will be at the bottom. And indeed, plastics that are floating on the surface of the ocean are vulnerable to UV radiation constantly. And this indeed is a great factor in the natural, let's say, degradation of the bigger plastics into microplastics. So yes, very perceptive of you.
So the next question is by Melanie, and she's asking, to which proportion do microplastics depend on their origin or on the destruction process?
Could you please just clarify what you mean to which proportion? Do you mean in terms of a certain type of microplastic comes from a certain place or contaminant, or something of the sort? For example, we know a lot of rubber contamination comes directly from tires in cars. Any of us who have been around the car know that the point of tires is to degrade but let the car go forward, and we eventually have to replace them. So I'm not sure I'm answering your question perfectly. Maybe you can clarify in that regard, but not only are there certain groups of plastics that are particular culprits, as rubber, as I've mentioned, but also in different strata of the ocean, so surface level or even benthic, we can find different types of plastic particularly. So you can find many, of course, but there are some that are more expressed. And this is, of course, also due to their density and the way that they find themselves around nature, how they're carried, even through wind currents if they're small enough. So yes, it does depend in that regard. Right.
Then we have another question out of box from Ander Martinez. Then he's asking if you can actually use turbulent flow as a friend. So there are viscosity, density, and other parameters involved in the Reynolds equation. Could you actually engineer them in a way that you can actually use turbulent flow instead of laminar flow as a kind of filtering technique?
Mm-hmm. So Selim, maybe you can help me with this if my answer is not ideal. So what I would say is that in a very rudimentary way of looking at it, laminar flow allows us to have control, more or less, of where the particles go, and in terms of outlets, and also in how they move in the microchannel. Another point is that turbulent flow in microfluidics, while possible, can also be difficult because, for example, you increase the velocity, but sometimes, depending on your channel, of course, especially if it's a longer channel with a high resistance, you can enter the field of very high pressures. And so this also evolves into a materials engineering, point of view. Is it possible to control turbulence? I'm not sure you can control turbulence per se, by definition, but I'm sure there might be some application in which turbulence can be useful to cut a laminar flow or to manipulate your fluid in a certain way. Selim, do you have anything to add?
Yeah, I mean, it's the definition, right? So it doesn't matter what you call it. So it's we call it turbulent flow if the Reynolds number is larger than 2000. So it's not like having turbulent flow is not gonna like help you to separate your particles. So it's just the definition. What we can control is the channel dimension, flow rate, and this, when you make your engineering according to this, you're automatically below 2000 Reynolds number, which is like laminar flow. So it's not like, it's just the name, it's the definition. So it's not like having advanced turbulent flow for separation. It itself doesn't mean anything. It's just the name. So what we can do as researchers, we can just play with the dimensions of the channels, width, height, depending on your application, depending on the particle size, and you can have higher flow rate or lower flow rate, and then you try to separate. So we shouldn't just stick to name what is turbulent or laminar. We should just consider what our application requires.
Right. If there are no more questions, okay, Andrew is thanking you. If there are no more questions, I can ask one. So in your research, what do you see as the biggest barrier in your way to get microfluidics and microplastics working together and in order to actually manipulate them in the way you want? So what's your biggest challenge right now?
I would say the answer is different between me and Selim because we're working with a bit different technologies. In my case, I would say that I'm working with acoustics at this moment, and I would say that as you go down to nano, my barrier becomes that while I can actuate big particles, so I can divide them from the rest, as I go down, the power, let's say power is not the correct word, but the wave that I have to put through my device into the microchannel increases in frequency and in energy. So a bigger dampening exists at the edges of my microchannel and inside my fluid. And this goes into the realm of something that's called acoustic streaming, which creates basically a vortex inside the channel. And this vortex is difficult to model and control. But there's a lot of work being done in that regard. But to answer your question in one sentence, it's simply the transition from micro to nano is very complicated in terms of technology development. That's why it's important to attack it from many different angles. In my view, Selim?
Yeah, it's the same for me. I mean, I'm specifically using inertial microfluidics at the moment, and when you look at the equations, it's micro world and nano world is very different. So when you have power of 4 of some particle size, this is if you go from 1 micron to 50 nanometer, this power of 4 is like dramatically changing the system. So that's why you cannot, at the moment at least, simply think microplastic separation and nanoplastic separation for the same system. So this is the main challenge at the moment.
Yeah, perfect. And I think Daniel is raising his hand. Daniel, if you unmute yourself and ask your question.
All right. Thank you, Martin, for your presentation. It's been quite insightful and reminding me of the physics I kept running away from in school then. Yeah, I just want to ask, when you get this method established and you're able to do separations and you get those nanoparticles or nanoplastics, don't you think you need like a chemical reaction-based test to substantiate the fact that what you've extracted is nanoplastics? Because the nanoparticles can also come from different sources that are not necessarily plastics. How do you intend to take care of that?
Mm-hmm. This is a very good question, and it's especially relevant when considering the next presentation, where we will start to talk more about spectroscopy and the endpoint analysis of our project. but it's simply a matter of using, well, spectroscopy, Raman techniques that are able through, let's say, in a very roughly speaking, using light and lasers, they're capable of taking a spectra that tells them exactly what type of plastic it is, what's the size of it, and with this in consideration, we can analyze and validate our systems. And of course, we have collaborations with many different types of institutes and companies that help us in portraying a better picture of our systems.
Thank you.
You're welcome.
Can I say something?
Sure,
maybe also Clementina can also say what's her challenge, because Martim and I talked about our project, but also she's doing something different. So it would be nice to hear. Yeah.
Clementina, please.
Sure. My challenge is developing a method that can actually be validated. So we were actually saying, someone said that before in the previous panel, that there's a lot of literature, but it's very hard to compare one paper from the other, and it's very hard to take some data as good because there's no actually validated method. So when we talk about real analytical validation, we have some parameter like the reproducibility, the repeatability, the accuracy of the method that it's necessary to assess. So that's why I'm here. My challenge is to try to show to the world that we can actually reach a method that it's reliable and robust for these kinds of analysis. And then of course analyze some food with it so that we can all know how much plastic are we actually eating.
Yes, so it's a sort of standardizing the methodology for food and water safety, if I'm correct.
Yeah.
Okay, that's brilliant. Thank you guys so much for your sharing and for your presentation. It was amazing. So if everybody is okay with it, we're going to go into the break until 15:05, so only 15 minutes, and then start with the second session. Thank you very much.
Olha, para no outro, na outra. Para, Jobs! Para, eu tô acabando de comer, Olivia, isso não tem faz, só me bate. Para.
Asked, are you here? Hey, Mehrdad, Yusuf.
Can you hear me?
Yes, yes, yes. Asked,
I
can
managed
you
to
launch
found
the
the
poll?
shade.
Nice.
Should we launch the poll?
Yeah.
Guys, is my background so noisy or is it okay?
It's okay for now. Yeah.
I'll keep myself muted until I'm done.
So.
Meanwhile, let's start from the first one. I will ask everyone just if you can choose the option.
Is it okay now, Asked?
Yes, it is going. I will just do you see still the poll?
I don't see it because I'm the host. Mehrdad, can you see the poll?
Okay, whoever is answering, it's
yes,
fine.
I can see the poll, but not the options to choose anything. Actually, I can't choose any option.
Okay. We'll
okay, then let's finish because then we will start the second one.
Okay, Yusuf, the second session is yours.
I finished it, and should I share the results? No.
Arash?
Okay, we can.
Yes, Yusuf?
I think Mehrdad was asking you.
Okay, so thank you for filling this. Let's go.
Yep. Yusuf, the floor is yours.
Yes, yes. 1 second. I just need the program. So I'm on my phone now. That's a little bit of a problem, but it's gonna be okay. Okay, hello everyone, and welcome to the MonPlas Workshop. On the second part of the event after the break, we are mostly going to focus on characterization methods of microplastics that are being used in this project. As the first presentation, on the second part, we are going to have Mehrdad, Lotfi, Choobbari presenting the group that works on Raman analysis of micro and nanoplastics. So I know many of us here that I know personally are working on Raman spectroscopy, so I'm pretty sure that it's going to be very, very interesting for every one of us. So please, Mehrdad, if you can share your screen. And as always, if anyone has any question, please leave it in the chat or in the Q&A session after we finish off with the questions in the chat. I will let you raise your hands. Mehrdad, the floor is yours.
Thank you, Yusuf. Can you see my screen?
Yes, but if you would go on presentation mode, that would be much better.
Now it's on the presentation.
Now it's perfectly on the presentation mode. Perfectly.
Good. Thank you.
Please continue.
Good afternoon, everyone. I would like to start the second part of the workshop, which, as my colleague mentioned, is on the Raman analysis of micro and nanoplastics. First, let me introduce the ESRs of this presentation. On the left side, you can see Aisha Bibi from Austin University, who is the ESR 1 within MonPlas program. She received her Bachelor in Information and Communication Systems Engineering and her master degree in Photonic Integrated Circuits, Sensors and Networks. Then you can see me, Mehrdad, Lotfi, Choobbari, it's my name. I am working at Ottevaere University, Brussels Photonics team. And within MonPlas, my project is ESR 7. I received my Bachelor in Electrical and Electronic Engineering and my Master in Electrical Engineering, Nano and Microelectronic Devices. And next, you can see Shiwani, who is working at Leibniz Institute of Photonic Technology. She is ESR 10 within MonPlas program, and she received her Bachelor in Physics and Electronics and her Master degree in Optoelectronics and Optical Communication from IIT Delhi.
As you can see, and here I am the presenter of this presentation. Let's see what is the agenda of this presentation. First of all, in the first section, I'm going to talk about the principle of Raman spectroscopy and give you a brief introduction to this technique. Next, in the second section, I'll talk about a Raman analysis of micro and nanoplastics, and I will show you how Raman spectroscopy can be used for the analysis of micro and nanoplastics. And finally, in the third section, I'll talk about the ways that we apply these techniques in our PhD research project within MonPlas program. First, let's see what is Raman spectroscopy. Well, when a monochromatic source of light, or in a simple way, a single color source of light hits a molecule, the photons which make up the light can interact with the molecule and being scattered. Initially, it was believed that the photons all will be scattered with the same energy as the incident photons, which is called a Rayleigh scattering phenomenon. But later in 1923, a scientist called Adolf Schmeckel postulated the inelastic scattering phenomena, in which the photons during the interaction with the molecule can gain some energy or lose some of their energy. But after that, 5 years later, Sir C.V. Raman experimentally demonstrated this phenomenon, and it led him to win the Nobel Prize of 1930, and that this phenomenon has its name from this scientist. What we do in Raman spectroscopy is to plot the intensity of these energy shifted photons with respect to the incident photons on a graph like this, which is called the spectrum of this molecule or the sample under test. This spectrum can be very practical, and it can give us lots of information about the material under test. For example, it can give us information about the composition of the material, the state of the stress and strain, the crystal symmetry and orientation, the quality of crystal, and also the amount of material. All this information can be obtained by looking into different properties of the peaks within the Raman spectrum of that material under test. The next thing which I would like to mention here is that Raman spectroscopy is a very weak phenomenon, because typically 1 in million to 100 million photons hitting the molecule can undergo Raman scattering phenomenon, and the duration is very short, on the order of a femtosecond. But fortunately, there are different methods which can be used to enhance the Raman signal. Here in the next slide, I'll mention some of them. It's not minimizing, okay. The first method which I'll talk about is stimulated Raman scattering, which can be used to enhance the signal of Raman scattering. In this method, we use two sources of light to excite the molecule, and we adjust the energy difference between these two sources of light in such a way that this energy difference matches the energy difference between the rotational vibrational frequency, rotational vibrational states of the molecule with respect to the ground state. And therefore, this can stimulate the process of Raman scattering and enhance the Raman signal up to 10 to the power 5 in comparison to the conventional Raman spectroscopy. The next method which can be used to enhance the Raman signal is called coherent anti-Stokes Raman scattering, or CARS, in which again we use a pump light to promote the molecules into a vibrational rotational state. And after that, we use a probe light to perturb the molecules being already promoted into a vibrational rotational state. And therefore, here we use the anti-Stokes signal. And it can be enhanced up to 10 to the power 6 in comparison to the signal obtained by conventional Raman spectroscopy. The next method, which is very popular for enhancing the Raman signal, is called surface enhanced Raman scattering, or SERS, in which we take advantage of surface plasmon resonance to enhance the local electromagnetic field in regions typically called hotspots. And these high electromagnetic fields can in turn intensify the light matter interaction and therefore increase the Raman signal up to 10 to the power 11 in comparison to the signal obtained by conventional Raman scattering. And finally, we have a tip enhanced Raman scattering here, or TERS, in which we use a plasmon active tip and we bring it into the close proximity of the sample. And when we illuminate the lights onto the tip's apex, the local surface plasmon resonance will happen on the tip's apex and to some extent onto the sample. And it can again intensify the light matter, or to confine, and it can confine and enhance the light field and therefore enhance the Raman signal up to 10 to the power 6 in comparison to the conventional Raman signal. The next thing which I would like to talk about here is the instrumentation which are necessary for performing a Raman spectroscopy. Here you can see the 5 main categories which are usually used for performing or making a Raman setup. We have excitation source, then sample illumination, light collection, wavelength selector, and finally photodetector. Each of these parts has its own consideration and discussion, which I cannot go into the details because of the lack of time. For example, just considering the excitation source, it can be a narrow band laser operating in UV, visible or near infrared range. And choosing the proper wavelengths quite depends on your sample and experiment situation, the situation of your experiment. For example, here you can see the Raman spectra of a material obtained by 3 different excitation wavelengths. You can see that by having a short excitation wavelengths, for example, 532, the green curve, the green spectrum, the intensity of Raman signal is high, while we have a large background, which covered the specific peaks of the Raman spectrum of that material. By going into longer wavelengths, for example, 785, the red spectrum, you see that the features of the Raman signal or a Raman spectrum of that material has been appeared. On the other hand, the intensity is lower. By going even to the longer wavelengths, for example, 1064 nanometer, you see that the background is almost 0, while the intensity is also decreasing. So you can see how it is important to consider your sample, to consider the situation of your experiment, and to choose the proper wavelengths. The same is true for other parts. For example, the sample illumination, the light collection, they should be quite carefully designed to help us to efficiently illuminate our sample and also collect the Raman signal. As I mentioned, the Raman signal is very weak, so here we need a really, really nice collection optics to help us to measure the Raman signal very effectively. Here you can see at the right side of the screen a schematic of a typical arrangement which can be used to implement Raman spectroscopy. You can see the laser light coming out from the laser, which will be reflected through a beam splitter, which can be a diachroic mirror, for example. And then we have the excitation optics, which help us to efficiently illuminate the excitation light onto the sample. Here, the Raman signal will be generated, and then it will pass through the collection path. Here we have the collection optics, which help us to deliver the Raman signal into a spectrometer. And in the spectrometer, we will use a grating to spread out the different frequencies. And finally, we will detect them using a CCD detector. And then it will go for further analysis into different software. Okay, this was a brief and general overview about the principle of Raman spectroscopy. and how this technique is working, and a brief introduction to its physics. Now, in the next section, I'm going to talk about Raman analysis of micro and nanoplastics. As my colleague mentioned, microplastics are ubiquitous in our environment. They have been found in marine water, wastewater, fresh water, food, air, and even the air we breathe. And unfortunately, researchers have recently demonstrated that microplastics revealed in the placentas of unborn babies. Or in another study which has been published in Nature Review, the authors demonstrated that these plastic bottles can feed babies by swallow millions of plastic particles per day, which is a huge number. And of course, this is not the end of the story. When this baby is grown up, he or she is still in the exposure of the consumption of plastic particles by different ways, like a food chain. And the bad news is that without any action, scientists estimated that there will be 12 billion tons of plastic in landfills and environment by 2050. But how should we tackle this problem? What is the first step? If you look into the different reports published somewhere, for example, from World Health Organizations, you will see that the first step is to have a better understanding of microplastics using quality assured methods, which can provide information about the number, shape, size, composition, and sources of microplastics. As my colleague mentioned again, for the analysis of microplastics, we have three main steps. First is sampling, which can be done from different sites like sediments, water, etc., from air. Next, we have sample preparation, in which we have, again, different methods and steps to prepare the sample and actually to properly filter out the plastic particles and prepare them for the next step, which is sample classification and identification, on which my research and some of my colleagues' research are mainly focused on this part. Again, in this part, we have different methods for the classification and identification of plastic particles, such as visual inspection, micro FTIR, micro Raman, and other methods such as pyrolysis, gas chromatography, mass spectroscopy. As I mentioned, a good method should provide information on all these 5 factors, which are shape, size, number, mass, and composition of plastic particles. But as we will see, it's not possible to obtain all these information with only one method. For example, visual inspection, which can be done by a naked eye or a microscope, can only provide information about the shape, size, number, and nothing about the mass and composition of the plastic particles. Next, we have these thermoanalytical method, pyrolysis, gas chromatography, mass spectroscopy, which can provide information on the mass and composition, but nothing about the shape, size, and number of plastic particles. And finally, we have these 2 vibrational spectroscopic methods, which can provide information about the shape, size, number, and composition of the plastic particles, but nothing about the mass. The next thing, which is very important regarding each technique, is the size limit of detection. For example, you see visual inspection, as expected, can provide a size limit of detection on the order of 500 micrometer, while micro Raman provides the smallest size limit of detection, which is on the order of 1 micron, and even it can go below 1 micron in some circumstances. But why is it important to consider these parameters, the size limit of detection? To better understand it, let's look at some findings from some works which have been done already in this field of research. In one study in 2018, the authors investigated the presence of microplastic particles in bottled water, and they have targeted the particles larger than 100 micron. And they used the Fourier transform infrared spectroscopy to analyze these particles, and they have found a range of 0 to 66 particles per liter in these bottled water. In another study at the same year, the authors used Raman spectroscopy to analyze the plastic particles present in drinking and bottled water. and they found a range of 0 to 6,292 particles per liter. You can compare this number with 66 particles. And with 95% of detected microplastics smaller than 5 micron. Here, actually, I don't want to make a firm conclusion or to say this study was better or this method is better. The point that I want to mention here is just we should consider the sample, and based on the sample, we should go for a proper method for our analysis. And here in this image, you can see the types of the plastic particles found in this work. You can see that surprisingly, they have also found plastic particles in glass bottles with 78%, again, smaller than 5 micron. Okay, now let's see how do they do these kinds of analysis of microplastics in different matrices. One of the most popular methods which is implemented using Raman spectroscopy to analyze plastic particles is Raman imaging, in which, for example, we consider an area of interest and then we measure the Raman signal pixel by pixel, for example, through raster scanning. And wherever we find a Raman signal related to a material, then we attribute that pixel with an arbitrary color to that material. Here you can see an example of this method. In one study, the authors used this, this technique, Raman imaging, to analyze plastic particles in different situations and also in the size range from 1 micron to 5 millimeter. They investigated microplastic particles such as polystyrene, polyethylene terephthalate or PET, polyethylene or PE, polyvinyl chloride, PVC, and polypropylene. Here you can see an example of the result reported in this work. Image I shows a white light camera image of some plastic particles mixed together. You can see that it's not possible to distinguish the types of plastic particles from each other. And then they performed Raman imaging on these areas shown by a red dashed color. And after that, you see that in image G, it's easily possible to distinguish the types of plastic particles from each other. For example, the red color is showing polypropylene, the blue color is showing PVC, the green color is showing PE, and so on. As I've mentioned, they also investigated another situation where they mixed the plastic particles with sand, and you can see that it's possible to distinguish the types of plastic particles from each other and also from other substances. Okay, this was again a general and brief overview how Raman spectroscopy can be used to analyze plastic particles in different situations and different matrices. Now, in the third section, I'm going to talk about how we apply Raman spectroscopy in our PhD projects within MonPlas program. Okay, the first project that I'm going to talk about is my project. As I've mentioned, Raman spectroscopy can be used for the analysis of plastic particles. But to have a reliable and accurate result, the analysis is currently limited to this bulky instrument and also to the clean environment such as clean room. Actually, we have to avoid contaminants, and also, actually, if we want, as I mentioned, to have a very nice result, we should use these bulky instruments. Here in the right image, you can see miniaturized Raman-based systems that has been developed in my group, Brussels Photonics team, by one of my colleagues, in which he has tried to reduce the size of the Raman system and also integrate a microfluidic chip with that Raman setup. This will be the approach that we will pursue during my project, and above that, besides that, we will also use other techniques such as diffuse reflection spectroscopy and scattering. These two techniques will be used to measure the concentration of plastic particles. As I mentioned already, it's important to get a sense of the number of plastic particles in the drinking water. Therefore, we will use these two techniques, scattering and diffuse reflection spectroscopy, to measure the concentration of plastic particles, and we will also use Raman spectroscopy to identify the types of plastic particles. And we will combine and integrate all these techniques with a microfluidic chip in a structure like this to perform our analysis. The next project that I would like to talk about here is Shiwani's PhD research project. In her project, she is aiming to develop a Raman microspectrometer. Raman microspectrometer is actually a combination of microscope plus Raman spectrometer. She's aiming to develop a high-resolution Raman system which provides which has a large field of view and help us to to measure and efficiently the Raman signal from our sample under test. Here you can see a camera image of the setup that she has recently developed, and she's also working on the automatization of the Raman setup through an intelligent software. The next project and final thing that I would like to talk about here is Aisha's PhD research project. As I've mentioned, Raman spectroscopy may suffer from some challenges like fluorescence background, low intensity of the signal, and to some extent it can be destructive if you won't be careful about choosing the power of your laser. From these challenges, Aisha is mainly working on the low intensity problem of Raman, and from the methods that I've explained before, like TERS, SERS, CARS, or SRS, she's focusing on surface enhanced Raman spectroscopy, or SERS. Her target is to develop a fast system which can specifically detect micro and nanoplastics by a flow-through plasmonic sensing system. And she will also perform angle resolve SERS using metallic porous membranes. And she expects to be able to detect the size of particles, the composition of the particles. As I mentioned, Raman can easily provide this information. And finally, she wants to also detect the morphology of the plastic particles, which is another important factor in determining the characteristics of plastic particles. Okay, this brings me to the end of my presentation. To conclude it, we have seen that Raman spectroscopy is a very, very practical method which can be used for the analysis of microplastic particles. It provides a very small size limit of detection. It may suffer from fluorescence, low intensity, and possible destruction of the sample. However, as I mentioned, again, it provides a small size limit of detection. So it does worth to work on these challenges. Next, we have seen that it's a crucial need to be able to perform online monitoring of plastic particles. And to achieve this goal, we have seen that optofluidic chips can be a nice option, the thing that I'm working on it during my project. And finally, we have seen that large field of view systems can be very helpful for measuring the Raman signal efficiently and also identifying the selection of the points of interest during the analysis. Thank you for your attention. I would also thank the funding of this project, which is from the European Union's Horizon 2020 under the Maria Skłodowska-Curie Innovative Training Networks. And also a special thanks to B-PHOT student chapter, which provide for organizing this event and also providing the opportunity for us to talk about our PhD research project. Thank you very much.
Thank you so much, Mehrdad, for your great presentation. Now I will welcome Aisha and Shiwani to join you as well to make the panel for the questions. We have already one in the chat box from Florian. So he's asking regarding Ms. Shiwani's PhD project. What will the goal be with regard to existing micro-Raman spectrometers in the market already? What is the resolution expected, the spectral range, parameters like these?
Hi, Florian. Thank you for your question. In my PhD, we thought of existing Raman microspectrometer also, but they are quite costly. So we are developing our own, and moreover, they don't provide you a very good field of view. We are also thinking of adding more modalities like fluorescence and phase contrast rate. So we are aiming to achieve very good field of view. Currently, we can have 3.2 into 2.2 millimeter field of view rectangle. Yeah. And you are asking about resolution also. We can have 2.4 micrometer result. Is that answer to your question?
Yes, almost. So if I well understood, it's mainly the main reason you are working on this project is that because the existing micro Raman system are costly, are quite expensive.
Yeah.
Okay. And you are making the goal of your PhD is to make a system only dedicated to microplastic or only microplastic. So you will create also your own database.
Not on database.
Okay. Yeah.
And from our experience, we know that Raman signal for plastics is quite high.
Yeah, but it depends. After, if you are talking about microplastic in water, microplastic in milk, in coffee, the matrices are always different.
Yeah.
Maybe you will also be limited by your CCD.
CCD. Yeah.
Because sometimes we have the limit of maybe around 10,000, maybe 15, 16,000 particles. This is the maximum we could see. So that's why I was asking what is the benefit of manufacturing a new system.
It's due to cost and with high field of view, because in market you can't have very good field of view system.
Yeah, you will use a dark field or
not, it's a bright field camera.
Okay. Thank you.
Thank you for your question. So I don't see more questions in the chat box, so I'm gonna go on with my own. So you're talking about the numbers of microplastics, nanoplastics inside different water sources, for instance, like water bottles, as Mehrdad said. But what's the standard technique that people are using right now to find these micro and nanoplastics? Like, you're giving us numbers, and that's your point, maybe to make it faster. But could you please shed some light on the standard techniques that are used right now?
Thank you, Arash, for the nice question. Actually, for the moment, one of the big challenges is that we don't have a standardized method for the analysis of plastic particles. Your question is actually referring to the biggest challenge at the moment in this world, which we don't have a really nice method to provide these kinds of information. As I've mentioned, because of this, we need a method which can provide a lot of information on the composition, size, number, morphology, and also mass, to be able to develop a standardized method for the analysis of these plastic particles. But if we want to get a sense about the number that you mentioned, well, most of the papers I've seen are mainly using Raman imaging. And if the particle size are large, then they will go for FTIR. And if we want to know the mass of the particles, then we should use thermoanalytical methods such as pyrolysis, gas chromatography, mass spectroscopy. Does it answer your question or not?
Yeah, I can see the point of the whole thing. Yeah, thank you very much. So another question. How will you solve the possible issues of overlapping different samples in the flow? So, for instance, you have different substances that are flowing through your microchannels. What would you do to, actually solve this problem?
Well, it's also a very good question, and also a challenge in my work. Well, for the moment, I think the only solution would be to use the microfluidic techniques that Martim has already talked about, to be able to properly separate the plastic particles from each other, and then pass them through our laser light and be able to detect them.
All right, perfect. So one more, do you see any issues related of wetting and floating the microplastics in water while doing measurements in the lab? So do you see any issues with this wetting and the fact that your microplastics might have a lower density than water and then rise up to the surface of water?
So far I use polystyrene samples because they are the only plastic particles which are commercially available in different size ranges. I have analyzed those plastic particles, and there are some special solvents which can be used to distribute the plastic particles evenly in the solvent. I don't remember the name of that solvent exactly. I think it's a kind of sulfates, etc. I don't remember it, but it's used to distribute the plastic particles evenly without having the floating problems.
Right, perfect. So another question, what would you do about the different autofluorescences that each of these different substances have? Because I think different types of polymers and plastics have different amounts of autofluorescence, which might be very harmful for actually measuring something Raman related in regards to them. So how would you overcome that difficulty?
Indeed, to overcome this problem, actually, besides the fluorescent signal from plastics themselves, for real samples, there might be some external exotic particles attached to the plastic particles, which can rise a lot of fluorescence. Well, to overcome these problems, I have measured the Raman signal of some plastic particles. The best way for the moment was to increase the wavelengths of the excitation source. For example, at the visible regions, we had a lot of fluorescence, but by going into the longer wavelengths, such as 785, the fluorescence was very low. And we are using that wavelengths currently.
All right, very good answer. I'm out of questions and I don't see anybody else putting questions in the question box. So any last remarks from any of the panelists? Any challenges you want to point out? Any futuristic ideas that you might have? Aisha, Shiwani, if you guys have anything to say, to add? No? All right, then we can move on to the next group. Thank you guys so much for your presentation. The next group is going to talk about detection of nanoplastics using plasmonic effects. And this group has two ESRs, 13 and 8, Udit Pant and Natalia Andrea Gutierrez Andrade. Here you go. The floor is yours, Natalia.
Okay, thank you very much. Thank you very much for joining this session. So the topic of this session is detection of nanoplastics using plasmonic effects. Till now, my colleagues have mainly talked about detection of microplastics, but in this session, we will focus on the challenge and how we will tackle the problem of detecting nanoplastics. So first of all, I am Udit Pant. I am a PhD student in Queen's University, Belfast. I completed my master's from Indian Institute of Technology, Dhanbad, India. And my colleague, Natalia Gutierrez, she's working as a PhD fellow in Vrije Universiteit, Brussels. And she completed her master's in applied physics from Columbia. So let's talk about the limitations on the existing techniques. So we know that at the moment, we have two main type of techniques. First is based on microscopy, and the second one is based on spectroscopy. The main limitation with microscopy is that it gives you false positive as well as false negative results. And spectroscopic techniques have an advantage in that terms. Basically we use generally Raman, FTIR or mass spec kind of technologies, but still there are some issues with the existing spectroscopic methods for the detection of micro nanoplastics. So mainly dominant techniques are FTIR and Raman based techniques. And the main limit of detection is 10 micrometer and 1 micrometer. when we talk about the size limit, so respectively, and the signal intensity when we talk about the Raman, it gives Stokes and anti-Stokes lines, and which have very low intensity, as talked in the previous session, that only 1 photon out of 10 to the power around 8 or 9 photons goes into that region. And then the spectral database, if we compare it with the microscopic analysis, the spectroscopic analysis bit tiresome and time consuming. So these are the challenges at the moment. And in this topic, first of all, we will start by history of plasmonics, then what is surface plasmon resonance, and we'll talk about some technicalities, then we will talk about our projects, and finally we will summarize we have talked about. So let's start with what is SPR, what is plasmonics. So the ability of noble metals such as gold, platinum, etc., the metal nanostructures to manipulate light at the nanoscale. This has resulted in an emerging research that is called as plasmonics, and it exploits the term that is called surface plasmon resonance. We will talk it about a bit later. So let's talk about the history. The first documented work about plasmonics can be said to start from the middle of 19th century. The systematic interest in colloidal system began with Dr. Michael Faraday's work in his famous 1857 lecture on the topic, a Beckerian lecture on the topic, experimental relations of gold to light. And it was the first scientific investigation for the chemical synthesis of colloidal gold. And he described the optical investigation of very thin gold films of ruby red colored suspension and of ultra microscopic gold in various liquids. And if we see, this state of the art work was still there, involving the embodiment of tiny gold particles. And we could see as early as 4th century BC, in ancient Chinese ceramics, gold in its very small sizes were used to tune the light and look sharp reddish spots. And in 4th century AD also, in the very famous Lycurgus cup, which is currently exhibited in the British Museum, the glass cup is dichoric in nature, resembling a jade with an opaque greenish yellow tone in direct light, but turns to a translucent ruby color when light shines through the glass. I mean, when you see it through the transmitted light, you would see a different color. And when you see it to the reflected light, you see a different color, which is, in case of reflection, it is green. So actually, the chemical analysis suggests the presence of mixture of both gold and silver nanoparticles contributing to its essential colors. The gold component being mainly responsible for the reddish transmission and the silver for the greenish reflection. So then after, in 1908, Gustav Mie gave a theory by calculating the absorbance of colloidal gold particles as a function of particle size using classical electromagnetic theory. And then after, in 1925, Richard Zsigmondy received the Nobel Prize in chemistry for his work in the field of modern colloid chemistry by studying the properties of gold solution and inventing using ultra microscope for that. And after that, by utilizing these things in 1957 and 73, we could see there were two famous methods came, which is Turkowitz and Franz respectively. And these are the benchmarks that still we are using for making gold colloids.
So we have heard until now a little bit of plasmonics and their effects seen through history, but what is plasmonics? So plasmonics are based on this phenomena called surface plasmon wave. Let's see a little bit more about this. So we know about the electromagnetic field, electromagnetic waves, which are an electrical and a magnetical field propagating through space. And this one is the behavior of light and also some other frequencies in the spectrum that we know already, such as infrared and also microwaves or UV light. But there are other interactions of electrical field and also that are also interesting. For example, the polariton waves. Polariton waves are waves that occur inside dielectric materials, and they happen because there is a reorientation of charges of the inside the molecules of these materials that will orientate according to an external electrical field. So if we apply an electrical field such as the one in an electromagnetic wave, then the charges inside the dielectric are going to move following the same pattern. Also, we have plasma oscillations, which are similar to the polariton waves, but occur inside in mediums where electrons are free, such as plasma or metals. And here we can observe a little bit of this, where the electrons are oscillating inside metal nanoparticles according to an external electrical field. But what happens if we combine these three effects in one? This occurs in an interface between a metal and a dielectric. So what happens is that light is absorbed by the electrical charges inside the metal, and this will generate a plasma oscillation, and the displacement of the electrical charges of the plasma oscillation will generate an electrical field inside the dielectric medium, which will also cause a polariton wave, as we can observe here in the graphic. What is important here also is that there is a penetration depth inside the electrical medium, and this depth is going to give us the range where we are going to be able to measure inside the dielectric. And this depth goes around, it goes between 200 nanometers up to 10 micrometers, depending on the wavelength that we are using to excite the plasmons. But if you think in our daily lives, we don't see this because, for example, in mirrors or also in metallic surfaces, we see a shiny surface instead of a black one. So what's going on? And what happens is that we require special configurations to be able to generate these effects, such as this one that we are observing here, which is the prism configuration. And also we require that the wave vector of the light it's equal to the wave vector of the surface plasmon wave. So even if we have a broad light source like this, we will observe that there is only one small gap that is going to be transmitting its energy to the surface plasmon wave. And this is due to the specific characteristics of the mediums that we have of the metal, which can be a specific metals such as gold, silver, aluminum, or copper, and also of the dielectric medium that we are measuring, specifically the index of refraction of this medium. Besides this, these techniques also enable us to use a binding layer, which will able to attach, for example, to our target compound. And when there is this attachment, then there is going to be a change of the index of refraction, which could be detected in the system. So this technique will provide us a fast method of detection, also a very high selectivity, and allows the detection of submicrometric particles.
Okay, so till now we have talked about how plasmonic helps in SPR based detection. Now, what happens when we combine this plasmonic effect with Raman system? Then it is called surface enhanced Raman spectroscopy. So Fishman and his group from the University of Southampton, UK, they were the first group. They reported the enhancement in Raman signals in 1974. Although the enhancement was seen, but how it is happening was wrongly interpreted until 1977. The precise mechanism responsible for the enhancement observed in SERS is still very controversial as found in the literature. There are mainly two mechanisms which are responsible for the large enhancement. of weak Raman signals obtained from any analyte. So first of all, Zenmeyer and Van Duyn first proposed a theory based on electromagnetic effects and which was responsible for the enhancement of Raman signals. This is known as the electromagnetic theory of SERS and is based on the excitation of localized surface plasmons. Albert Chent and Crichton first proposed the theory based on the charge transfer effect of the adsorbed molecule on the surface of the substrate, substrate molecule on adsorbed molecule for the Raman signal. This is known as the chemical enhancement. There are two kinds of enhancement, but the dominant one is electromagnetic enhancement, which helps to enhance the signal from 10 to the power 6 up to 10 to the power 10, 12, 14 times. And it is because of the plasmonics. Let's talk about what are some other applications of plasmonics. In medical terms, we see generally plasmonics generally helps to for drug delivery related applications in medical sciences or cancerous treatments. And in terms of electronics, it can be helpful for fast optical communications based systems. For food security, it works for the detection of any kind of contaminant in the food if it is present. And mainly in photovoltaics, we could see there are some examples of plasmonic solar cells, which helps to enhance the sensitivity, which helps to enhance the efficiency of the solar cell. So we have some commercially available systems for generating surface plasmon effect and helps in achieving our detection. Like similar to other techniques such as Raman and FTIR, there are also other commercial based techniques. One of them is called BioPur, and this system provides the optical unit and microfluidic sample delivery system to perform measurements with SPR. However, to be able to measure binding events, the user requires a sensor chip, which he must functionalize to achieve the binding layer for the measurement. This system has a multi-channel to be able to perform different measurements at the same time. There are also high throughput systems such as BI 4500 and that are based on SPR imaging, which enable them to have many measuring spots in which the intensity changes indicate the amount of binding events of the target molecules. Now, let's just talk about our projects in MonPlas and how we will deal with the nanoplastic detection challenge. So in my topic is to develop a rapid and low-cost platform for the detection of micro and nanoplastics. So for that, I'm going to work on surface enhanced Raman spectroscopy, since Mehrdad has talked about what are some limitations and challenges, and one of them is that the intensity is very high when we deal with Stokes and anti-Stokes, and SERS gives better replacement in that terms, that it enhances the sensitivity manifold. So these substrate helps to enhance the signal, and these can be of different types based on hard substrates and based on other nanostructure based SERS enhancement substrate. And basically, after developing that, I will work on to incorporate these substrate into a microfluidic chip. So that will help to rapidly detect these nanoplastics, and giving the signals from Raman, it will help to chemically visualize these nanoplastic as well.
In my project, Surface Plasmon Resonance for the Detection of Micro and Nanoplastics, I'm going to develop a prototype of a sensor based on a surface plasmon resonance technique, which integrates also a microfluidic system, a functionalized binding layer, a freeform lens design, and all for the detection of micro nanoplastics. But you may ask, why do we need all this? So SPR chip is the detection side, but it also requires aid of all these other parts to be able to perform as a whole sensor. So first, one of the goals is to develop a binding layer that will selectively attach to the polymers that we want to detect. Then we also require a microfluidic system to be able to deliver the sample into the SPR chip. And also we require an optical setup that will provide the light, the illumination, and also the detection phase of the system. And we want to include a freeform lens because this is a method in which we can reduce the number of optical elements in the system that will enable us to have a smaller and portable device to do in situ measurements. Also because this freeform lens design will also enable us to reach a lower limit of detection, better performance of the sensor. So I want to mention you one of the challenges that we are facing in our projects. In SPR, one of the main challenges is the binding, functionalized binding layer to be able to selectively target the polymers that we aim to detect. And also in general for the sensor, we expect to reach a low limit of detection, very good repeatability and high selectivity. In the case of a ESRs project, there are many goals. First, some of the challenges are the ones that are faced usually in Raman spectroscopy, such as the fluorescence background, accepting the limit of detection size. Also, Udit aims to develop a device with fast processing and a substrate able to attach to the microplastic samples that he wants to detect. Finally, I would like to give you a brief summary of the topics that we have mentioned through this presentation. First, that plasmonics-based techniques can fill the current gap in micro and nanoplastics detection as they overcome other techniques' limitations. Second, plasmonics can be used in diverse configuration and sensing mechanisms that allow it to have a wide range of applications. And finally, MonPlas projects, in MonPlas projects, plasmonics will be used for microplastics detection through different techniques, such as SPR and ESRs. Finally, we would like to show you some of the references that we have used in our project through this presentation. Oh, okay. And would like to thank our funding, Marie Curie Actions and the European Commission, and also the B-PHOT student chapter for organizing this event.
Thank you so much, Natalia and Udit. It was an amazing presentation. So let's go to the questions. So firstly, Olga is asking, what functionalization procedure do you use to attach plastic particles to the metal surface?
Well, this is quite a difficult question, but we have also seen a little bit about it in my project, because usually the target molecules in SPR are proteins or enzymes that have very specific 3D structure that enable them to bind selectively to another molecules, but which doesn't happen with polymers. So for this, we are basing not in the usual, we are trying to aim for not the usual method of functionalization layers in SPR, but there are also other methods such as molecular imprinted polymers, which also are based not only in the general, like the binding sites of the target compound, but also in the geometry of it. So this we think would help us to selectively attach to the polymers that we want to detect in the future.
Brilliant. So another question to both of you, Natalia and Udit. So what aspects, what parameters can you think of that affect the sensitivity, and what parameters do you think would affect the sensitivity and the stability of your SPR and SERS-based sensors?
Natalia, would you like to start with SPR? Then I'll talk about Raman, space SERS. Hello? So,
I think she's muted.
Okay, so let me start with myself. To fabricate a SERS substrate is also a challenge, because at the moment the techniques we have is basically electron beam lithography or ion beam lithography and these techniques. And one challenge is the reproducibility of the substrate that you are fabricating and how big substrate you can fabricate. So for making a low-cost substrate is a challenge, because in the project we are going to develop a low-cost system, and there are some techniques like 3D nano imprinting techniques and these techniques. But the problem with that is, again, generally to bake this kind of substrate we use polymer. So in order to detect polymer, we are using polymer. And so it may contaminate the sample and the substrate. So these are the challenges that we need to face. And other than that, the intensity of light you are using, these also depend upon the substrate structure. Like if you deal with the gratings, it has the SPR wavelength at a particular region. So if you have the wavelength in that region only, then you will get a good enhancement. So these are the existing challenges with the substrate.
Oh, okay. I guess I'm invented now. So in my case, for example, SPR has different configurations and also different interrogation methods that achieve, each one of them are capable of their own limit of detection. So in our case, we are aiming to use an interrogation system based on polarization or interferometry, which enable us to have better approach and also to the number of binding events, and also to reduce the noise background that are usually present in the optical systems, such as the dark current noise in the detector and also the instability of the light source.
Perfect. We have Olga with a raised hand. If you want to unmute yourself and ask your question.
Yes, thank you. Thank you guys for the nice presentation. I followed my question to Natalia and maybe to biochemists in the group. So do you think that, I know it's not in the scope of your project, but do you think later it will be possible to detect plastic particles in the blood samples if we know the biofunctionalization procedure? Do you think it will be possible?
Well, I must say there are two steps. First, we aim to have a preparation of the sample phase before entering it into the SPR chip. So in this phase, it is possible to remove some of the particles or blood compounds that could be interfering with the SPR signal, I think. And also when they enter the SPR chip, the binding layer, if it is able to achieve high selectivity, then it will also filtrate the type of particles that are present in the sample to selectively attach to the ones that are of our interest in the detection process.
Thank you, Natalia. Good luck with your project.
Thank you, Olga.
All right, let's go to Tatev.
Thanks a lot for this nice overview on plasmonics. I would like to ask the question about the cross-contamination, because Udit mentioned that there is a problem of contamination if you use like polymer substrates from SERs, but I mean, you also need to do the metal coating, right? So what are the other sources of cross contaminations, and whether there are also solutions to kind of get rid of these cross contaminations? So the question can be addressed to both. Thank you.
So I would like to start with this. So in a few days back, I was reading a literature. In that, actually, the substrate they made were of polymer, but then they deposited a metal thin film so that this possibility of getting contamination can be reduced. I'm not saying that it can be achieved 100%, but the possibility that the contamination may be there can be reduced by this method. And other than that, as we see that plastic is everywhere, and Martim presented in his presentation that that plastic, when you are taking the sample from the micro pipettes and all these things, these are made up of plastics. So we should take care in every step of our, from sample preparation up to the detection part. And because everywhere, even the system that you are using may have these kind of elements that may contaminate your sample. So we should take care of these things. Better to have clean rooms, and as Martin mentioned, that that clean rooms, wearing gloves, even the coat you are wearing may have a potential source of contamination for that. So these kind of things, I think we should take care while working on these environments.
Also, I'd like to add that we must be very careful with the type of material that we are incorporating into our systems, because as you mentioned, there could be possible cross-contamination. So in this way, in this approach, the best idea would be to use a different material than the one that you are targeting. And also, if it's possible, to replace it with glass for the freeform components and microfluidic system.
All right, thank you, Tata, for your question.
Thanks a lot. And if you let me, like Udit, I think you can also reduce the cross-contamination by using, as Natalia mentioned, the glass pipettes.
Yes,
also, they exist. Yeah, and it is just as advice, but I guess this can really reduce the pipetting, the tubing, everything from glass. Microfluidics can reduce the contamination part.
Thank you. Thank you for the advice.
Perfect. So I don't see any more questions in the chat box, but maybe I can ask one myself. So Udit, you're working with nanostructures, but then you want to actually detect microplastics. So my understanding is that the nanostructures got to be like close to each other and have a certain type of morphology so as to make the type of hotspot that is needed for CES. So what is your solution to fitting these microplastics inside those?
Actually, my aim is to detect nanoplastics.
Oh, okay. All right. So they fit, like when you say nano...
They can be, they can't be. So the thing is, you need to design your substrate like that, depending upon the target you want to achieve. Suppose, and I mean, I was telling about, it's not only because of the particle size, it's because of the light you are using, because at the end of the day, we need to generate this SPR effect, and that is totally dependent upon size and shape of the structure that you are having. So that's why.
So Olga is leaving a comment. Thank you, by the way, Udit, for your answer. So do you consider phase measurements? They might increase sensitivity.
Yes, this is a question for me.
Oh,
yes,
okay.
I was mentioning earlier that there are different interrogation schemes, such as wavelength, angular, and intensity. But besides this, there are phase interrogation schemes, such as heterodyne optics, also interferometric system and polarized system. We are aiming to use a polarized system or interferometric system because it's possible, they are possible to miniaturize and be used in portable systems. However, their limit of detection, like the highest limit of detection that they achieve, is up to 10 to the minus 7, 10 to the minus 8 power. So this is our purpose.
Right, perfect. So I don't see any more questions, and you're already at 4:14. So I propose we move on to the next group. But meanwhile, if Asik is here, she's gonna actually initiate the polls. And yeah.
So I will just run now the polls for 10 seconds. If you can, please answer them.
Yes.
The first one.
I don't think anything has popped up for me, but Natalia, Udit, are you seeing anything?
Yeah, people are answering already.
Okay, okay.
Yeah.
So it's only gonna take 10 seconds. Meanwhile,
okay,
I'm just gonna...
Sorry, I will just end this one and I will launch the second one. Okay. So here are just the results. That people are interested. That's why they join to know more about microplastics. Yeah, and the second one, very quickly, we want to know from where did you know exactly about the event, because most of the people replied that they would like to know more about our upcoming events. Was very nice for us, and so we want to know how we can share more, let's say, the information for the general public. Okay. Some more seconds. Okay, thank you.
All right, thank you so much, Ast. So let's go to the final group who's going to talk about micro and nanoplastic analysis through spectroscopy and imaging. There are ESR 2, 5, and 9 here. Syed Atif Iqrar is going to be the first presenter. Then we're going to continue with Jordi Valls Conesa as our second presenter, and finally go to Julia Boeke. So,
hello, can you see my screen?
Yes, yes, we can see your screen, hear you loud and clear. Thanks.
Oh, thank you. So, hello everyone, my name is Syed Atif, and today I'm going to present about, this is the final presentation of the workshop, and we are going to present micro nanoplastic analysis through spectroscopy and imaging. So here comes the today's presenters. First is Julia Boeke. She's from Leibniz Institute of Photonics Technology, Germany. And she's ESR 9. And secondly, this is me, ESR 2 Syed Atif Iqrar from Western University. And the final presenter is Jordi Valls, and he's from Bruker Optics, Germany. So this is the agenda of the presentation. Firstly, I will go over the PL spectroscopy, introduction to photoluminescence. Then I will discuss about the potential of photoluminescence for microplastic detection. And then we'll move over to the FTIR and QCL spectroscopy, where Jordi will discuss about the IR emission, FTIR QCL system, and then have hyperspectral imaging for microplastic classification. And finally, Julia will talk about imaging flow cytometry. She will talk about optical detection methods, introduction to flow cytometry, and multispectral imaging. So this is, so I will just start with the photoluminescence spectroscopy. So before going to technical details, so first look and understand what is photoluminescence. What is the principle of photoluminescence? So photoluminescence is quite simple. It is basically the emission of electromagnetic radiation when a material is exposed to light. And there are two types of photoluminescence. One is fluorescence, and the second one is phosphorescence. And the basic difference between them is the time. And fluorescence is known as the immediate emission of radiation after the absorption of photon, and for phosphorescence is the delayed emission of electromagnetic radiation after the absorption of photon. So here you can see in an animated image, a photon comes, it gives energy to the electron, and then electron jumps from ground state to the higher energy state. And then when it comes back, it emits another photon. This is the simple phenomena of spectroscopy. And here you can see for layman audience to understand what is happening, I included a animation analogy in which ball is coming, this is relevant to the photon is coming, it is giving energy to the man, it is going, then the man is going up to the building, and when it is coming back, it like emits the photon. So this is like the basic phenomena of photoluminescence spectroscopy. And as you can see here in the diagram, the light is absorbed. When it comes back immediately, then it's called fluorescence, and if it takes time, then it's called phosphorescence. So here are some like practical examples of fluorescence spectroscopy you can see in the images of different in biomedical industry and chemical industries. So today's topic is like to discuss that how we can detect microplastics using the photoluminescence spectroscopy. I would like to mention here is that PL spectroscopy is quite new in this field, as Raman and I are, there are a lot of work done on in this field. But in terms of PL spectroscopy, it is quite new, and many of the researchers, they are trying to explore the potential of this technique for the detection of microplastics. So here is the latest research done in the latest paper published in year 2020, and they use the excitation wavelength of 405 nanometer, and this is the basic PL experimental setup they used, and you can see there. You can see that they use the laser diode of 405 nanometer, and then they put an excitation filter. Then the light laser goes and it like reflected by the diachroic mirror, and then it goes to the sample, and when it reflects back, so we then using the spectrometer we plot the emission spectra, and we see the different spectra of polymers and non-polymers. So in this experiment they use different 9 polymers, plastics, and 9 non-plastic elements, and they like found that it is possible that PL spectroscopy, it has potential to detect plastics. So they found that they found the emission spectra, and they found the different wavelengths of PL. Furthermore, they like plotted the spectra, but the problem they faced is like it's really difficult to differentiate between different spectra. So like for that purpose, they introduced in the 2 parameters that are basically intensity ratios, and they use that 2 intensity ratios, and they have taken the 3 intensities actually, the value of the value of intensity is at 3 wavelengths. The first one starting from 427, this is the minimum wavelength, and the maximum is 552, and they have taken in between them, and they like plotted another graph based on these 2 parameters to differentiate between plastic and non-plastic materials, and they were quite successful in differentiating plastic from non-plastic using this PL spectroscopy. And also the same group, they like they published another paper recently in 2021, and this time they used the excitation wavelength of 266 nanometer, and they also claim that it is like non-destructive and fast technique, and they use the commercially available macroplastics. They actually, the problem with this technique is that we are not like sure about the resolution of this technique, and still we are like trying to explore whether this photoluminescence can be a replaceable technique for detection of plastics or not. So in this case, they like got different spectra, and they use the same methodology of defining 2 parameters, and by this time they also differentiated different plastics from non-plastic materials. This is like another paper which is very important one, and this time they used staining protocol like to increase the intensity of the signal. So this time they use Nile red as a stain, and they use the same condition as previously with the 405 nanometer LED, and the samples were stained at 60 degrees for 10 minutes in a 50 microgram per milliliter, and they performed several studies, and the purpose of this study was to investigate to get the optimized or standard protocol for the Nile red staining. So in the picture below, you can see the setup they use. It is quite similar with the previous one, and they use the solvent acetone and ethanol. And in the first, they use ethanol 100%, and in the second experiment, they used acetone ethanol in a 50-50 ratio. And they got the different spectra of different 4 plastic particles. And also they perform the experiments with different temperatures, with different concentration of nitrate and at different times. And they finally conclude and optimize the protocol for the red nitrate staining for microplastics. And they concluded that the nitrate concentration of 20 microgram per milliliter, staining time 10 minutes, and ethanol acetone mixture of 50% is the best and optimized protocol for the detection of microplastics. So this is like a very recent area, so we don't have much like much thing to discuss about it, but there are some challenges we are facing with the PL spectroscopies. Real spectroscopy is the first one is incrustation, and you know that when we deal with the real samples, the plastics are incrustated by the organic materials, and it's really difficult to get the spectra of these of the same material when it is like covered by different organic materials. And the second problem is the dyed plastic products, and the other one is the photo bleaching is another problem, because we are dealing at UV range, and it's like cause photo bleaching. So these are basically the recent challenges the researchers are facing to detect microplastic using this photoluminescence technique. So thank you so much. I will like hand over the presentation to Jordi. So Jordi, could you please?
Hi, thank you so much. My name is Jordi. I will talk about FTIR and QCL spectroscopy methods and imaging using them. I'll mainly talk about infrared emission, FTIR and QCL, and imaging. First of all, we have to consider that infrared light is invisible radiation with longer wavelengths than visible light. More specifically, in QCL and FTIR, we will work on the mid-infrared range. And this also implies that the energy of infrared photons is lower than from visible and UV, meaning that instead of working with atomic emission, we will work on molecular emission. For that, in terms of infrared absorption, molecules will absorb infrared light, and this vibration will result in a dipole momentum change, making it to absorb certain wavelengths, depending on the types of vibration they are doing, either stretch or bending. These wavelengths depend on the strength of the bond, the atoms involved, and the type of stretching or bending. And thus, a spectra will be generated for each kind of molecule, each spectra being different. This makes that for a different compound, each spectra will be like its own fingerprint, being able to be analyzed in further detail using infrared light. For infrared detection, when an infrared light incites a sample, it is absorbed with the procedure I talked before, and this absorption causes the amplitude of certain wavelengths to be reduced, mostly the ones corresponding to the molecular spectra. And then some kinds of light will be emitted, mainly on reflection, transmission, scattering, and fluorescence emission. But I will be centering on reflection and transmission. There is also another detection technique that is called attenuated total reflection that is used both in QCL and FTIR and works like this. An infrared light source enters an ATR crystal and incites a sample, usually above their critical angle. So this light will be totally reflected, but since it's in contact with a molecular sample, it will absorb the certain wavelengths according to the molecular spectrum. This also means that the reflection infrared light will be absorbed. I have no control over the presentation, I'm so sorry. This means that the infrared light will be detected by the detector, and comparing this totally reflected light with the infrared source, we will be able to measure the infrared absorption. Now, in terms of the first detection method, it is Fourier transform infrared spectroscopy, or FTIR, and it basically is the use of an interferometer. This interferometer is made using a beam splitter in combination with two mirrors, one of them with moving components. That basically what it does is to split the light between two, and then this light comes back to the beam splitter to be collected. Firstly, to be absorbed by the sample, and then to be collected by the detector. Since this light is interfering with itself, and this moving mirror changes the parameters of the light, it will cause an interferogram for the light. But as you can see here, this interferogram is not readable by us, and thus we have to apply a digital Fourier transform in order to obtain a spectra of the frequency of the material and analyze their molecular components. These are some examples of Fourier transform infrared spectroscopy. As you can see here, we have an image for visible light and an image for infrared light of a PET microplastic sample. And the thing is that this infrared image is not only a 2D image, but it's also a 3D image, because every pixel of the image contains the whole spectra of the pixel measured by FTIR. This certain image is called a hyperspectral image. Now, in terms of the second detection method, we use a microscope that uses a quantum cascade laser as an infrared source. Quantum cascade lasers, or QCLs, are semiconductor lasers on the range of the mid-infrared. They are composed by two main regions: an active region, where the photon is emitted by an electron lowering its state, and an injector region, where this same electron goes using tunneling through a lower region of an active region to the upper region of a new active region. Thus repeating the cycle. This process of a single electron emitting multiple photons raises the name cascade on the QCL. Also, QCLs are made of thin layers of semiconductor material in a periodic pattern, alternating between active and injector region. In terms of QCL systems, they are basically made of a combination of different QCLs with different tuning. This means that they are built differently in order to have different spectral ranges. This way, when we put all of them together, we have a larger spectral range that is that can be comparable with the one on the FTIR. As you can see here, the light, the infrared light, goes basically through the sample and then is collected by a camera, either by transmission or by reflection. As you can see, the light is directly absorbed, and there is no need of any digital transformation, unlike FTIR. And unlike FTIR, we are able to measure single wavelength images, because in FTIR we are only able to obtain the whole spectrum. Although in QCL we are also able to obtain the whole spectra using a sweep scan. And finally, in terms of microplastic imaging, using those hyperspectral images obtained by QCL and FTIR, we can use an imaging method in order to classify and detect the microplastics inside the sample. To do that, we use a machine learning algorithm that is trained using microplastic spectra in order to classify each pixel of the image as any kind of microplastic. After that, we use another imaging method in order to segment the particles independently to be furtherly analyzed. Also, this method can be done using a hyperspectral image that is basically the whole spectra, or a hyperspectral image that uses multiple single wavelength images as a source, the ones coming from the QCL. And now my colleague Julia will talk about another imaging method relating to visible light.
Thank you very much, Jordi. Yeah, so as you've seen from all the previous presentations, we talked a lot about different optical detection methods. And therefore, I would like to start with a short summary. Yeah, here you can see basically the range, like the resolution for which those techniques are more or less applicable. And so you see that we have already heard like a lot of them today, especially when it comes to FTIR and Raman. And what we see is that here, especially for the region around 1 micron, 10 microns, it's actually getting quite low when we see what methods we can use for that size range. Obviously, we can also use pyrolysis and JCMS, so gas chromatography, but this is unfortunately a destructive method and it's not optical. So yeah, we discard it, but I just put it in because yeah, it's one of the very few that actually can also go down to nanometer, nano-sized particles. However, when we look at what we want to measure, then we also have to take into consideration the technique we want to measure it with to make sure that we don't get biased. Because, for example, yeah, when we look at especially microplastics that tend to get smaller and smaller over time, because now we also recently discovered that there's not only microplastics, but there is also nanoplastic particles. The method that we use to detect these particles can be largely dominated by either small particles or many particles at all. So therefore, we have to choose the method carefully. During these next few minutes, I would like to introduce you to a combination of optical detection methods. The presentation got stuck, yeah? Can you still see it?
I still see the diagram that you had for different techniques.
Yes.
Something purplish has appeared on it.
Yeah, exactly. Right? Yeah,
okay,
that's totally fine.
Thanks.
You're welcome. So what I would like to introduce to you today is the combination of the microfluidic technique that already got mentioned so many times earlier, and now some of these optical techniques. And if we merge them together, we reach something that's called imaging flow cytometry. So imaging flow cytometry is, okay, there was one slide missing. Yeah, sorry, I'll just explain it to you. So imaging flow cytometry basically is a very old and already proven technique when it comes to cell analysis. So what you should see in this picture technically is this small stream of particles. Let me highlight it for you if I can see the pointer. Yeah, here it is. So you see this channel here filled with particles, and typically here on the top you would have a laser source, and then here you would have a photomultiplier collecting the scattering from the particles. So it's a very reliable technique with high throughput to analyze cells. So you're able to assess the size from the scattering and partially their shape, but yeah, only, as I said, partially. The main limitation is that you basically don't gain any additional morphological information or any information on the spectrum. What is then done is that that you can additionally stain these particles with fluorescent dyes. So there it comes into play that you then would implement linear variable filters in front of your photomultiplier tubes as the detectors. So you could also collect their spectral characteristics and try to determine basically the material composition in terms of fluorescent signals from your samples. So basically similar to what we've seen from Atif earlier. And now if we would basically get rid of this imaginary laser that we unfortunately can't see right now, but now replace it with this microscope setup here, we unfortunately lose a bit of the throughput, because here you see that we are only now limit ourselves to about 5,000 cells for this method. But we gain information, because now we are actually also able to see the particles, and we are able to assess their shape and their size more precisely. And this is what we're going to now use further, because as you've seen, we've already gained so much more information. Of course, we lost throughput, but if you compare it with other techniques that are now on the market or in research labs to analyze microplastics, then 5,000 in this terms particles per second is still quite huge when it comes to microplastic analysis. So we're still happy so far. And what we are now going to do in the next step is, and this is where we then dive from standard imaging flow cytometry to where the innovation of my project comes into play. We apply a linear variable filter over our channel. So what we do is we use an broadband LED source, and what we then see is basically a color discrimination of the particles. And luckily, microplastic particles itself are often colored, because they degrade from plastics, which often are already colored themselves. So it's a very nice approach in which we can make these plastic particles visible. Let's see what we see on the next slide. Does it come? Yeah, we've already been there. Sorry, guys. Yeah, so one of the main drawbacks, though, from general microscopy, some of you might already know that, is focusing. So when we want to look when we actually want to see our particles, which is what we have to when we want to implement an imaging technique, we have to make sure that all our particles are actually in the focal plane of our setup. And as I said in the beginning, we're now using microfluidics. So basically the particles are passing through our channel, and so far we don't really know where they are. And this isn't good, because everything that is not in our focal and is not in our focus, we can't see it. And this is exactly where the microfluidic chip design comes into play. So what some of my colleagues developed is, I actually like this technique a lot because it's kind of smart. So what they did in the beginning is, here you see the entire chip, and now we're going to focus on the blue region. So here your sample will go in here, and here you have a sheet flow that ends up in this flow focusing unit. So you have a first part where all your samples will be, where all your particles will be nicely aligned before they enter this staircase region. And the staircase region is actually a rotation unit. So what comes out of it is all particles perfectly aligned in our focal area. And what we then can do is we compress them a bit more so that we can use the nice laminar flow that Martim introduced in the beginning, and have all our particles in our imaging region here, which we have encoded with the visible filter, because mostly microplastics are colored by nature already. Well, not by nature, by human, but as they are you know what I mean. And when we now illuminate them, you will actually see a brightness difference depending on their absorption and transmission characteristics. So when you have a very dark particle, you know that it has a very high absorbance in that region. And when you have a very bright particle, you know that for this wavelength, the particle is basically becoming transparent. So if we now apply this to real life samples, I brought you some colored pollen here. So we actually stain some of the particles, and when you now look at this group of multicolored particles that are now flowing here through the channel, you see that they actually change color depending on where over the filter we collect the image from. So basically now the positional changes that we can collect from imaging and their gray values directly reflects the spectral properties in the defined wavelength range. So we are now able to basically gain the shape information of the image, as well as the spectral information and and additionally, we could also acquire part of the phase objects and the internal structure of particles. For microplastics, that's less relevant. So thank you very much for your attention. And we are all happy to answer your remaining questions. And of course, huge thanks to B-PHOT and to the European Commission, who's so kind and funded us.
Thank you so much, guys, all of you, Jordi, Julia, and Syed Atif. Thank you for your presentations. Now, guys, we are open to questions. So if you have any questions, you can raise your hands. And if you are feeling shy, you can put it in the chat box. I'm going to ask it for you. All right, I'm just going to start with the first one myself. So, Jordi, you said that you're using different algorithms to actually detect these different microplastics in an image. So what algorithms are you looking at right now?
Well, mainly I'm using random forest for as a machine learning algorithm. It is basically a classifier. What I'm giving it is like thousands of microplastic spectra. And then what it's able is to analyze another spectra and tell if it's what kind of particle that it's already trained of is.
And for it, you have to actually have a database or a sort of library, right?
Yes, yes.
And do you acquire them yourself, or?
Yes, I acquired them myself by now.
All right, all right. And what does that say about the generality of the technique? So, for instance, in the end, in the final application, you're going to use it for, I don't know, actual water from the sea and so on. So then what happens to the generality? Is it only good for specific types of plastics that you anticipate to be there?
In principle, it should be good for any kind of plastics, mainly because I'm focusing that the algorithm learns the most common plastics first. And also I'm working with it, but I plan for it to work on every kind of sources. Yeah, not only some kind of water. Even we are talking about working with tissues, but for now it just works. We're working with the water part.
Thank you very much. I can see Ast has her hand raised. Ast, can you ask your question yourself?
Thank you, Aisha. So I just would like to thank all the presenters for a nice presentation. And my question is mainly for Julia. So you just mentioned that you are going to use LED with linear variable filter, because in that case you will be able to detect, let's say, all the different colors of plastics. So why do you need to use, let's say, linear variable filter for that, or like what is the resolution that you need? Mm-hmm. And to be able to detect, let's say, each color, and what are the power requirements for that?
Very good question. We are still currently working on the limits of the setup. So obviously in the beginning, you would ask yourself, why would you need that when you have an RGB camera, right? But it's not only like the aim is not only to to sort colored particles themselves, but also to later add a chemometric analysis. So if we then stain the particles and want to color discriminate them, then the resolution of the filter actually becomes huge, because when we look at the RGB camera and how the pixels are composed, then yeah, you only have like 3 different values, which are then shaped together as gray values. So you lose a lot of information there, and you can't really relate that to a spectrum that you acquired from a reference from a spectroscope. So yeah, therefore we use the filter because it gives us the additional information while still keeping the image.
Yeah, so you just use the filter to, let's say that only some certain wavelengths will pass and you will be able to detect exactly that one. So yeah, based on your experience, maybe do you know already which are the most commonly used like wavelengths? So you told that they are from 450 till 650 nanometers?
Yes, like we use visible range right now because we aim to use dyes that have absorbance and in that region. So obviously we will not detect fingerprint because it's just not in that region. But that's why we use dyes and staining to discriminate the particles. And then it depends a lot on the characteristics of the dyes and the chemistry of the plastics on how they come together. So it's difficult to ask because you can actually like tailor it towards the filter so that it's applicable. But I'll hope to give you more information on that next year. Currently, I'm still too much in the beginning of that part of my project.
Okay, good luck.
Thanks.
So we'll be happy to see the results.
Yes, for your question. So, is there any more questions? All right, I'm going to ask another then. So you showed the part of the microfluidic channels that were like in steps. What is the point of rotating these droplets, I assume? Why do you want to rotate them?
We want to rotate the particles because when they are entering the channel of the microfluidic, like when they enter the microfluidic device, then they are spread over the entire channel. So we have a width of like several tens of micrometers and also height of several tens of micrometers. So the particles can be anywhere over that entire channel. So we use this staircase unit to basically change their flow direction to make sure that they will be perfectly aligned in the focal plane of the microscope setup.
All right.
So that all of them will be imaged sharply.
Yeah, but if they are inside the droplets with a
no, they aren't.
They are not.
No, they are just flowing through a liquid.
Wow, and then
like
these steps would make them into small. Okay.
No, like the particles are in aqueous solutions.
Yeah.
So basically by changing the flow, as similar to what we saw earlier, where in the presentation where we had the differentiation between laminar and turbulent flow, like, and I'll say it very rudimentarily, but if you add a drift towards how the fluid flows, then you can actually change where the particles would arrive. And so we basically use this the staircases to add like tiny bits of turbulences all over the chip to make sure that all of them will most likely end up right in our focal plane.
Okay, yeah, very interesting, very interesting. All right, I can see a message. All right, if nobody has more questions, we're 2 minutes away from the ending of the Q&A. Then we can use these two minutes to actually turn on our cameras for a group picture, and then we're going to go to Professor Ottevaere for the ending of the session. So if everybody could please turn on your cameras and ask, is everything okay for the picture?
Yes, now I'm waiting until everyone switched the cameras. Yes, I'll just wait a few seconds until more people are appearing. And these pictures we are going to post in our social media. So if there's someone who don't want to have his picture or her picture, so please don't switch your camera. Okay. Okay, so ready? 1, 2, 3.
Should we be smiling?
Yes, preferably. And please wait a few minutes that I will take another one. Yes, 1, 2, 3. Thank you, everyone.
All of them taken?
Okay. Yes.
Oh, perfect. Thank you, Ash. All right. Thank you, Julia, Syed, and Jordi for your amazing presentation. Then we can go to Professor Heidi Ottevaere for the ending speech. Hey, Natalia, is there anything?
What? Oh, hey, yeah. No. I'm sorry.
Okay. No worries. Heidi, the floor is yours.
Okay, thank you very much, Arash. So in view of time, I won't keep it very long, but I hope everyone here present really enjoyed the very inspiring talks this afternoon during our MonPlas workshop. So we learned how micro and nanoplastics particles can influence our life, how we can manipulate them, and of course also how we can detect and measure them with a variety of different measurement techniques. So first of all, thank you very much to all our early stage researchers, so our ESRs, to share with us your knowledge, your work and your first results. So I really enjoyed it, and I hope the public present today also enjoyed it a lot. So I'm sure that it will be of very much of an added value for all people present here, and it will be very inspiring for future work, or at least for out of interest. It was very enjoyable to be present here. So in case any of you does have some questions, please, you can always contact the MonPlas Consortium. This can be through social media or also through the website, so the MonPlas website. So feel free to contact us in case of questions, some future collaboration, etc. And to end, I thanked already our ESRs for their very interesting talks, but I also would like to thank the organizers. Of course, I mentioned already the content was set up mainly by Mehrdad and Natalia, but we also had our B-PHOT chapter that was here today to, first of all, promote the event. And there I would like to thank Astrid and Timo, but then also for moderating everything and everything that goes around with the event. Thank you very much, Arash, as well. So you did really a very great job today. So, and by this, I would like to close this session, and I wish you a very wonderful evening and hope to see you soon.
Thank you. Bye.
Yeah, have a lovely day. Bye.
Thank you, Heidi.
Thank you.
Thank you very much for being here.
Thank you.
Thank you very much.
Thank you, everybody.